रेनू दिख रहा है?
हेलो, हाँ शैली, नहीं अभी तो नहीं आ रहा।
अच्छा, थोड़ा-सा लैग भी होता है उसमें ना, 1 आध मिनट
अच्छा।
का।
हम्म।
हाँ, मतलब उसपे जो इस पे आ रहा है ना ज़ूम पे वही दिखना चाहिए।
हाँ, तो वो लिंक से सीधा नहीं खोल सकते हमने? अच्छा, हमारे यहाँ से
आ
जा
तो
रहा है
रहा
या
है
नहीं है?
रेनू, आ रहा है, आ रहा है, आ रहा है। अब आ रहा है। आ गया, आ गया, आ गया।
कैसे पता लगेगा? वो लिंक ही यूज करना पड़ेगा?
हाँ, वो लिंक से हाँ, मैंने देख लिया रेनू, चल रहा है ठीक। अब हम जो बोल रहे हैं वो सारा वहाँ पे रिकॉर्ड हो रहा है, लाइव जा रहा है। गुड मॉर्निंग ऑल ऑफ यू। गुड मॉर्निंग डॉक्टर उत्कर्ष।
Good morning, ma'am.
आ गया, बट शैली उसमें थोड़ा लैग आ रहा है ना, जो मैं अभी
हाँ,
बोल रही थी
हाँ,
तब वहां पे सुनाई दे रहा
हाँ, वो थोड़ा-सा लैग आता ही
था। हाँ, ठीक है फिर तो।
आ रहा है वहाँ पे। तो कोई एक यूट्यूब चैनल पे भी थोड़ा-सा ध्यान रखें, वहाँ लोग चैट बॉक्स में लिख रहे होंगे कुछ-कुछ, तो और फीडबैक फॉर्म भी वहाँ पे जाना है। फिर देखना पड़ेगा ना, मैं यहाँ से छेड़ नहीं सकती। हेलो।
Hello, good morning, everyone.
हेलो सर। Good morning, sir. We have with us Professor Hemchand Jain, Principal Deen Dayal Upadhyay College. Good morning, sir.
Good morning. Good morning.
Recording in progress.
Hello, ma'am. Hello, Chandralekha, ma'am.
Hi, how are you?
I'm good, ma'am. So nice to have you here.
Thank you. I'm delighted to be here.
Yeah, the pleasure is all ours, ma'am.
Thank you.
We'll start at 8:35.
That sounds great. Thank you.
Yes, ma'am. Professor Ruplal is having some issues joining, so we'll just wait for a couple of minutes more. He's just joining.
That sounds good. Thank you.
Yes, ma'am.
मैं जूम पे हूँ. हेलो.
Good morning, sir. So can we start? Sir?
Let's start. Are you able to listen? Are you able to hear me?
Yes, sir. Yes, sir. We can hear you clearly. Yes. Yes. So, a very good morning to all of you, all our microbiology lovers, or I would say the lovers who are going to be the lovers of microbiology. So today on 17th of September 2021, I, Dr. Shelly Anand, on behalf of the entire organizing team of MICROSPHERE 2021, wholeheartedly welcome you all to this International Microorganism Day celebration, particularly our children from schools and colleges across India and abroad, microbiologists, teachers, principals, parents, and also the microbes because it is their day. So warm greetings to our respected delegates and principals of different colleges of University of Delhi and the collaborating institutes for joining us today on this special day. But why is it called as a special day? So you all must be wondering, why should microorganisms have their own day at all? Well, this is extremely important in the present times when the entire world is a witness to what a tiny virus is capable of doing. So, even more important now to raise awareness among our young fellows and the society in general on the essential role of microorganisms on our lives, and also as an occupation and career. They're important in all spheres of life, and this conclave will be a window to peep into the magical world of the microbes and their role around us. Now, before we move ahead with our program, I would like to invite Professor Hemchand Jain, Principal Deen Dayal Upadhyay College, to welcome our guests and speak a few words for our participants today. We feel pleased to inform you, sir, that nearly 900 participants from the breadth of our nation and abroad have joined us, and this event is being telecast live on YouTube also. May I request you, sir?
Thank you, Shelly ji. A very good morning to all for the opening session of the MICROSPHERE and International Outreach Program. I extend my heartiest welcome to our esteemed speaker, Professor Chandralekha ji from Department of Physics and Astronomy, University of Pittsburgh, Honorable Professor Rooplal ji, Nasi Senior Scientist, Terry, New Delhi, President, Principals of colleges, my respected colleagues, friends, and dear students. Today, we are here on a common platform to celebrate the International Microorganism Day as a MICROSPHERE, an international outreach program under our initiative to promote microbial literacy in the society and raise an awareness for the role of the microorganism in our lives. And also as a profession and career with the school students, undergraduate students, and teachers. Our colleague has been organizing several outreach programs in the past for the knowledge enrichment of these school students, both in rural sector and Delhi. To name a few, are the one was the Bunny School Innovation Camp, Basics 3 in 2018, outreach program for class 10 students of Kendriya Vidyalaya Sector 12, Dwarka in 2018, outreach program on Chapter 1 DDUC of Bayer Initiative is School Outreach Program for Enrichment in 2019. Outreach program Chapter 2 DDUC Bunny School Innovation Camp in 2019. Online international conference with a special session on reaching out to children and society a step towards promoting microbiology literacy, a virtual outreach program in 2020. This COVID-19 pandemic has made it even more essential for us to understand these tiny microbes around us. While some of these are fuse and cause disease, the majority of others are friends and contribute for the welfare of human and society. Uh, I wholeheartedly welcome and appreciate this collective effort of our college, Deen Dayal Upadhyay College, in collaboration with IIT Roorkee, Achar Nendradev College, Gargi College, Krodimal College, Maitreyi College, Ramjas College, Venkateshwara, Magad University, Allahabad University, and our industry associate FISGen Private Limited to make the students, teachers, and society aware about the significance of microbes in our day-to-day life. That's why we are celebrating International Microorganism Day today, that is 17 September. We will try to adopt the celebration of the International Microorganism Day as one of the good practice of our institute from this year onwards. I hope this program will help you to understand the importance of tiny microorganism for welfare of the society. My best wishes to organizers and thanks to all the participants who joined on this day. Thank you very much.
Thank you, sir, for always being so supportive. Well, students, on this onset, I feel like sharing an interesting tale behind this date, that is 17th of September. Now, anyone who has ever studied biology must have heard of Antony von Leeuwenhoek, the father of microbiology. Students, he was a Dutch merchant with no scientific training and little education, but grew up as a curious young man who loved examining things around him. His homemade lens and the microscope were his sole companion, and he kept on observing everyday things from corn kernels to fungi. So in 1677, when he observed the canal water through his homemade microscope, he was amazed to see what he called as the small animals. Usko usne animalcules kaha, the first microorganisms ever observed. He then sent a letter to the Royal Society of London with all his observations on this specific day, which earned him a place in history as one of the pioneers of the microscopic world. So this monumental letter was dated 17th of September, an event which is worthy of commemorating and acknowledging his contribution to science. This is why 17th September was chosen as the day to celebrate the huge impact of the variety of microorganisms that surround us. So today, under the mentorship of Professor Roop Lal, uh, who is the chief coordinator of this outreach program, and many more extension activities organized in the past, we have opened a new chapter in line with our ongoing efforts towards International Microbial Literacy Initiative. This global initiative aims at improving the general perception of people towards the microbes and make our children informed citizens of tomorrow. So, from food to industries, from the gut to the environment, these microbes have a deep relation and impact on our lives. So, while many use them synonymously with germs, it's time that we understand how majority of them are our friends. They are not always the foes. So today we celebrate these microbes as our friends and a step towards sustainability. So on this platform, when we have so many people joining us, especially our young minds, we would like to take you all through our journey of being closely associated with promotion of science and particularly microbiology in students of rural India. So one man's dream, helping transform the future generation. So here it goes, students. So I'll be sharing a short video.
दिनों से मन करता था और कई बार मैं बिना किसी को बताए स्कूल में आकर अपनी पुरानी यादें ताज़ा करके चला गया। तो वो जो नींव बना दी थी उन्होंने और स्कूल ने, वो मेरी जिंदगी में बहुत काम आई। वही नींव नहीं, जब दूसरी या तीसरी कक्षा में थे तो वो जब स्कूल मेरे नाना जी देखने आते, आप भी अपने माँ बाप को कहना है कि स्कूल आओ, हमें देखो हम पढ़ रहे हैं कि नहीं, वो चेक करने आते थे। और जब छुट्टी के टाइम मुझे दुकान में नीचे थी, न दूध प्याज न देकर जलेबियां। एक दिन वो आए और मैं स्कूल में भाग गया था एक दो बच्चों के साथ। लेकिन बयान हुए बड़े मारे काकड़ी लंबी हुई तो ये थी नदी गली। और उन्होंने देखा कि मैं क्लास में नहीं हूँ। मैं छुट्टी के बिल्कुल सही टाइम पे इधर खेतों में इधर उधर होकर सब कुछ चुरा के घर पहुंचा और वो आए। मैंने सोचा इनको तो पता नहीं होगा, इतने जोर का चाचा मारा उन्होंने मुझे, मेरे से सहन नहीं हो पाया। और फिर इतना ही नहीं, जो टीचर थे उनको कह के चले गए कि तुम्हारा बच्चा क्लास से नहीं जाना चाहिए। और दो बच्चे जो हम भाग के गए थे क्लास से, दोनों को पूरा दिन ऐसे बस यही करवाते रहे। उसी टाइम में मेरी कमर की दर्द अभी ठीक नहीं हुई, पूरा दिन कराया था उन्होंने। क्या बोलते हैं उसको, ये
उठक बैठक
मैंने पूरा दिन कराई थी। उसके बाद मैं स्कूल कभी नहीं छोड़ा, लेकिन मेरे मन में इच्छा हमेशा।
आज का अब यहाँ से यहाँ हम कैसे पहुँचे, खुदाने के लिए ढूंढते रहे। ये तो हाथों की मेहनत है, ये और भी मेहनत होगी। बताओ कौन सी मेहनत है? तो क्या आपको की तरह अब ऐसे मत कहीं पे पड़े हुए ले लेना, चोट खाने से जो दिमाग बढ़ जाता है, बड़ी जगह लिखा होता है। इसीलिए जो है कोने पे लिखा हुआ क्वेश्चन है, उसके बाद जो है कुछ अपने एफर्ट से या एनवायरनमेंट से भी होता है। एक पूरे देश का बहुत बड़ा स्पेक्ट्रम है। तो अगर आप लोग जो है जवाब नहीं दे पाओगे, चलो इस बार नहीं, अगली बार जवाब सभी आने चाहिए। पहली बार माफ कर दें क्योंकि किसी ने आपको शायद इतना चैलेंज किया ही नहीं।
आपने कभी माइक्रो ऑर्गेनिज्म को देखा नहीं थे, और क्या आते हैं माइक्रो ऑर्गेनिज्म के अंदर? आप लोगों को बैक्टीरिया का पता है, एक सेल होता है, वो उसके अंदर वॉल होता है, सेल मेम्ब्रेन।
आप दो चार्जेस को सेपरेट करेंगे। आप क्या क्रिएट कर रहे हैं वहां पर? यू आर क्रिएटिंग अ पोटेंशियल इलेक्ट्रिसिटी का जनरेशन, ठीक है? आठ को आपने कन्वर्ट कर दिया बाइनरी नंबर में, तो क्या करेंगे? जैसे आपका ये नंबर है, एक को कन्वर्ट करना है, सामने बनाना है, तो मुझे क्या करना चाहिए? अभी आपने देखा कि एक सेल बनाने के लिए क्या चाहिए होता है, बेसिकली करंट को फ्लो कराना है तो क्या क्या चाहिए?
अगर उससे हम मल्टीपल काम करवाएं, मैं उसे बोलूं कि साथ में एक कोई पर्टिकुलर एक कैलकुलेशन है वो भी करना है, साथ में कुछ दूसरा फंक्शन है वो भी।
किसी भी 1000 जीबी तक की बिना पॉसिबल है, ठीक है? कोई लिमिट बस उसका इंफ्रास्ट्रक्चर ज्यादा चाहिए। इस मशीन के अंदर भी इस मशीन की कैपेसिटी कितनी है? आप 50 से नीचे के कितने भी नंबर हो 50 की टेक्निक लगा सकते हो, 100 के नीचे कितने भी नंबर हो 100 की टेक्निक लगा सकते हो। पेपर में कभी भी कहीं पे भी किसी भी क्वेश्चन में ऐसा स्क्वायर करना हो तो करने की जरूरत नहीं है। अगर आपको ये टेक्निक याद है, बस फटाफट मन में सोचा और आंसर देख दिया।
ये भी फॉर्म क्या है? ये कैसे है? और ये कैसे नहीं मिलेगा? इजी है, तो बहुत चलता है ऐसा, है ना? बुक में वो बनाया होता है ऐसे करके, और बहुत सारे ऑर्गेनेल, माइटोकॉन्ड्रिया, फ्लो, सब कुछ दिख रहा होता है।
और हमीरपुर 2 डिस्ट्रिक्ट हैं जहाँ शौचालय और
जगह जाना, या मैं यहाँ पे चेक छोड़ जाऊंगी, या आपके मोबाइल, इसमें मोबाइल बैंकिंग नहीं होगी, उसमें भी IMPS का ऑप्शन होगा, तभी देखिए। तो आपको जोश से बोलना है और खूब हिम्मत से कॉन्फिडेंस से बोलना है। कोई नहीं बोल, बैठ जाते हैं।
कॉन्फिडेंस देखना है हमने, आपका मैटर देखना है. America counted its votes, then India had a sleepless night in counting their. पहली बात देख ले, हमारे माननीय प्रधानमंत्री जी ने नहीं पता कि मोबाइल के थ्रू पैसे ट्रांसफर
हर बच्चे को आगे बढ़ने की अंदर तमन्ना होती है, लेकिन वो कभी किसी को बता नहीं पाता. ये स्कूल बताया ऐसा नहीं था, यहाँ की सड़क ये ऐसी नहीं थी, यहाँ इतनी दुकानें नहीं थी, और आप जैसे तेज बच्चे नहीं थे. पता नहीं कैसे इसी समय में पैदा हो गए इतने अच्छे अच्छे बच्चे. हमें तो अचंभा होता है ना. आप बहुत अच्छे हैं, जैसे सारे टीचर बता रहे हैं कि आप हर प्रश्न का इतनी अच्छी तरह से जवाब दे रहे थे. और एक प्रश्न जो मैं बार बार शेयर कर रहा हूँ, जिसका जवाब मुझे बहुत अच्छा लगा, जब पूछा कि भारतवर्ष में सबसे ज्यादा क्या दिक्कत है, मुश्किल है. इसको भी नहीं पता, लेकिन इसने पता क्यों कहा कि अगर ये समस्या खत्म हो जाए तो हमारा कितना बजट और कितने पैसे बचेंगे जो हमें फ्रंट पे लगाना पड़ रहे हैं, जिससे बच्चों की पढ़ाई और सब कुछ हो जाए. तो जोर से बता अपना. मुझे सही जवाब चाहिए इसका. ऊपर लिखा है, ऊपर लिखा है लाइफ इज ए चैलेंज लिमिटलेसली. मैं हिमाचल की ली है, लेकिन ये हिमाचल को दर्शाने के लिए नहीं ली है. मैंने इसको अपने जिंदगी के अनुभवों से कंपेयर किया है आपको बताने के लिए. तो इस स्लाइड का ये माने है कि जिंदगी एक चैलेंज है. चैलेंज का हिंदी में कौन बता रहा था? क्या मतलब होता है? हाँ, कैसे बोले? अगर तो रूपलाल का बात करते इंग्लिश में कर दी.
Well, those were wonderful memories. So, if each one of us decides to go back to our roots, our school, our society, the world will become a better place indeed. So, this is a thought that the present generation needs to understand. Life is about giving back. Now, with that beautiful note, I would now like to apprise all our participants about today's program. Though it is not possible to cover all the areas where microbes have stood out as heroes, but we have decided to bring the best for you. Today we have amongst us Professor Chandralekha Singh ma'am, who shall be talking about on science and society and how imperative it is to promote science. Next we have Professor Ruplal, who will be talking about the microbial garden within your body, while touching upon the microbial literacy initiative, followed by Dr. Renu Solanki, who has brought some interesting tales on discovery of microbes and their products. Next, we have Dr. P.C. Ashwin Kumar with his concept of sustainable housing for future and how microbes can help decrease the carbon footprint. Then Dr. Amjad Hussain will be highlighting how the microbiology sector can be a route to entrepreneurships, followed by Dr. Utkarsh Sood, who will be briefing you about the intricate role of the microbes in achieving global goals and bringing in sustainability. Dr. Gauri Garg Dhingra and Dr. Roshan Kumar will be speaking on microbiology as a career. What are the choices after 12th and UG? Then we have specially planned a hands-on session with our young participants where our undergrads will show you how DNA can be isolated from your kitchen ingredients. And finally, we have an interesting quiz for the school and college students with cash prizes for the winners. So, does it not sound interesting? Yes, it does. So, without any further delay, let's proceed with the sessions. I now invite Dr. Princey Hira to introduce our guest of honor today, Professor Chandralekha Singh, ma'am. Over to you, Princey.
Thank you, Dr. Shelley. Good morning, eminent scientists, our respected principals from different colleges and schools, colleagues, and my dear participants. Today, I am pleased to welcome you all on behalf of all the organizers to the celebrations of this International Microorganism Day, MICROSPHERE. Now, it is rightly said that the aim of education is the knowledge not of fact, but of values. So it is on occasions like this when we are getting opportunities and we are looking forward to get a scientific exposure and interaction with our experts. We are very fortunate to have with us Professor Chandralekha Singh, who has been promoting science on various platforms. We appreciate your tireless efforts, ma'am, compassion and vision towards the well-being of the community. I feel privileged to welcome and introduce our speaker, Dr. Chandralekha Singh, who is a professor in the Department of Physics and Astronomy and the founding director of the discipline-based Science Education Research Center at the University of Pittsburgh. She's the past president of the American Association of Physics Teachers. She obtained her BSc and MSc degrees from the Indian Institute of Technology, Kharagpur, India, and her PhD in theoretical condensed matter physics from the University of California, Santa Barbara. She was a postdoctoral fellow at the University of Illinois Urban Campaign before joining the University of Pittsburgh. She has been conducting research in physics education for more than 2 decades now, and she co-led the US team to the International Conference on Women in Physics in Birmingham, UK in 2017. She is the fellow of the American Physical Society, American Association for the Advancement of Science, and American Association of Physics Teachers. Today, she would be speaking on how to enhance science by making it equitable and inclusive. We are very eager to hear from you, ma'am. Over to you, ma'am.
Thank you so much. I'm really delighted to be here. And can you see my screen?
Yes.
Okay, thank you. So, I wanted to start out by showing you a picture from my own discipline, physics. And if you look at this picture, this is a really famous picture that most of the people would actually know about. This is the famous Solvay conference. One thing that should stand out for you in this picture is that there is only one woman, Marie Curie, and that is about it. And if you look at a conference, this is a picture taken from a recent conference, you see that there aren't that many women here either. So the situation doesn't seem to be very different, so many years, like almost 100 years later. And so if this is the situation, we should be thinking about these kinds of things as teachers, so what do you think your classroom would look like to a student who's sitting in the 3rd row or 4th row and 5th row and looking at other students in the front? If nobody looks like them, would that somehow increase their anxiety about being in that class? And how might the students feel about their potential in that course just because nobody else looks like them, just because they don't seem to have any role model in science, right? And can the students' gender, their socioeconomic background, the fact that they might be first-generation college students, by that I mean like their parents may not have gone to college, right? How might those kinds of things affect how they're feeling in this class, right? And most importantly, what can we do as teachers, to empower our students and make sure that they embrace their struggles and use them as a learning opportunity and create a low anxiety environment in which all students feel like they belong, all students feel like they can excel, and all students want to work with other students. Because notice that if you don't feel like you belong in a class, you might actually not even want to participate in the class discussions and any other group activities that is taking place in that class. So the question is, what can we as teachers do to empower our students and make sure that everybody, and especially those people who are from these underrepresented groups, those who don't have role models, also feel like they belong? I want to point out something which is called stereotype threat. And stereotype threat refers to the fear of confirming a negative stereotype about oneself. So, for example, if there are not that many women in science, people don't see that many professors who are women. If they haven't heard of that many women, they might say, oh, maybe people like me are not supposed to be in this discipline. And in that case, if they are actually struggling with something, they might think, I'm struggling because I'm not as good as other people. What's the point of even trying? This kind of a threat that is associated with you feeling like you might actually belong to a group that is stereotyped, that is not supposed to be there for some reason or the other, can be very taxing. And the point is, cognitive science says that your brain can only keep 5 to 9 chunks, only 5 to 9 things at one point, no matter who you are. But if suppose some of those chunks, while you're thinking about the issues, when you're trying to solve problems in your science disciplines, are taken up by the anxiety of the situation, then you have, then that robbing away of some of your cognitive resources will leave even less resources for thinking about the problem and doing metacognition and doing reasoning. And that is not a good situation, right? So basically, we want to avoid stereotype threats. We want to make sure all students belong in the discipline. But notice that if suppose, you actually have some students in your class, and let's say that you say on the first day, oh, I know that, 10% of this class is not going to pass this course or something of that sort, then you can imagine that the people who are actually least, who have the least sense of belonging, for example, like people who are first generation college students, people who came from low socioeconomic background, right? I mean, those people might think that they are the ones who actually are not going to succeed there because you have already told them that 10% of the class is not going to pass this course. The point is, that is not something that a teacher should ever say, because our goal should be to actually make sure that all of our students excel. In fact, there was a recent study that showed that instructors who actually said that I really strive to make sure that every single one of my students does well, as opposed to having this predetermined feeling that only some people can excel. Those are the kinds of people who saw that there was no difference between the majority and minority people in their classes. On the other hand, the instructors who said, no, only like 30% of my class will do well, and all the rest of the people, I don't even know what to do with them. Those were the people whose students actually showed great difference based upon whether they belonged to majority group, whether they were already privileged, or whether they were students who didn't have the same privileges as other people. So our job as teachers should be to actually empower all students, as opposed to just giving opportunity to people who are already privileged and not other ones. Mansi, you have a question. Please unmute yourself and ask me. Did you have a question? Because I see you raising your hand. Okay, I don't see it. So what I was saying is, what I was saying here is that, in any kind of ambiguous situation, right, a situation in which people feel like, okay, I must be the person, I must be the only person struggling, or I will be the only person I'm the person who's going to fail. Those kinds of situations can be really taxing for people, and they can actually even force people to rely on stereotypes, and that can rob them of cognitive resources, and it can actually diminish their performance and lower their sense of belonging, and it kind of puts them in a negative feedback loop, and it can keep bringing them down further and further. So we need to recognize that we have a huge role to play in empowering our students. So what I would like to propose to all of you who are actually teachers is that, we for the longest time, we have thought of our role as being, that of dealing with students' offenses. When I say offenses, what I mean, we are trying to teach students effective problem-solving strategies. We are trying to help them develop useful skills like problem-solving, reasoning, and metacognitive skills. We are trying to help them transfer their learning from one situation to the other. We are trying to help them develop good conceptual understanding. But the point is that we should really be thinking of ourselves as coaches. And if you are a coach for any game, one of the things that you have to realize is that you cannot just think about offenses, you also have to think about defenses. So it's our job to basically strengthen both students' offense and defense. So what do I mean by defense? What I mean is students should feel like they belong in that particular community or in that particular classroom. They should have an identity as a person who can excel in science. They should have intelligence mindset that corresponds to growth mindset. By that, I mean, they should think of their brain consisting of muscles that can grow when they work hard and work smart, as opposed to intelligence being something that is immutable. Some people have it and other people don't have it. That is called fixed mindset. We don't want students to feel that way, but it's up to us as instructors, as teachers, to create that kind of a learning environment in which students actually have high sense of belonging, high identity as a person who can excel in science, have growth mindset, have high level of self-efficacy. That means their beliefs about whether they can excel in science. And the good thing is that if you can actually create an environment like this, then students will even become more interested in what they're doing, and they will even have higher achievement goals, and they actually will want to take risk. They would like to work hard and they would like to work smart. So the point is that making sure that students actually have strengthened their defenses also is really important to create a safe environment where all students, and particularly those from underrepresented groups, also want to participate in the community, or want to engage in the conversations and all of the other active learning things that you might be doing as a teacher. So when there is a good combination of both of these things, then your classes will be equitable and inclusive. So let me give you some examples from my own studies. First, I'm going to talk about self-efficacy, which is the belief in one's capability to succeed in a particular task, course, or subject. And it can impact students' persistence and engagement even after controlling for all of the other things, like controlling for their prior knowledge and skills, okay? And it can impact the effectiveness with which students engage in learning. And it can put them in that kind of a negative feedback loop that I talked about. So let me show you in our physics classes. So this is going to be data from my studies. And what I'm showing you on the horizontal axis is students' grade here. This is in Physics 1 course, and this is in Physics 2 course. And this is their self-efficacy in the vertical axis. And the blue thing here is for women, and the brown thing is for men. And this line is just a line that I'm showing to you that in Physics 1, women who were getting A grade had the same self-efficacy as men who were getting B, and things got even worse. And in Physics 2, women who were getting A grade had self-efficacy, which was the same as men with almost C grade. And notice that it's not even your grade that is the most important thing, but a combination of grade and self-efficacy. And if you don't have high self-efficacy, you might drop out of a discipline. You might not actually take a career that is commensurate with your qualification. And in that case, we will actually be compromising quality because of the fact that people didn't feel like, they were good at something, even if they were doing really well. Notice that also, if you think that you're not doing well, as I said, some of your cognitive resources when you're doing problem solving and when you're doing test taking would be taken up by the anxiety, and then fewer people would actually get A grade, which is what we are seeing in our courses. So the question is, what can I do? Okay, and here are some of the things that we hear women saying, for example, I have pretty low self-confidence when I see how other people are so smart, other people understand more than I do. in these courses. I feel like I have to work a lot harder to understand. I mean, it keeps going on and on. Notice that what is pluralistic ignorance is the fact that these women are thinking that they are the only ones who are struggling. The truth is that everybody is in the same boat. All the people even Einstein said, if you haven't struggled, you haven't tried to learn anything new. So struggling is the stepping stone to learning anything new. Students are in college to learn new things. We as instructors have to tell our students to be proud of struggling, to be proud of failing, to be proud of embracing their failure and learning from them and using them as a stepping stone to learning. So you have to tell your students, pat yourself on the back, be proud of the fact that you don't know something, because that is exactly why you are here. Otherwise, if you knew everything, then why would you be there? So basically, there is this field-tested approach to helping students develop high level of defenses. That means high sense of belonging and growth mindset. So we did one of these belonging mindset intervention that is actually an ecological belonging intervention. And by ecological, what I mean is that it actually is designed to change the kind of culture that you have in your classroom. It impacts the way students interact with each other and the way they interact with their instructor and the way instructor interacts with the students. And this is the great thing is that it's a very short intervention that you can do in half of your class. So in our case, first I'm going to tell you about a study that we did that we published last year. And in this, 3 instructors participated, and these were really large physics classes with more than 250 students each. And 6 recitation sections participated. By recitations, I mean these are small problem-solving classes in which you have maximum 40 students, and they work collaboratively in small groups, on problems. And we wanted to see how would students who do when they actually were in the social belonging group versus the control group. Control group means that those recitations did not have any intervention. So what happened in the intervention? We promoted the idea that struggling is the only way to learn something new. So students should be proud of struggling, since that means that they are already on their way to learning, and they should learn from their mistakes, learn from all of the things that they're doing, because that is not failure. That is actually part of learning. And even the facilitator, the instructor talked about how when they were in college, they were feeling the same way. They were embracing their struggle, because students appreciate it when you tell them about your own struggles. You tell them about your own resilience, because that actually normalizes struggle and it makes it, really real and normal for students. It will reduce their anxiety, reduce their procrastination, increase their enthusiasm for solving challenging problems, and ensure that their limited cognitive resources are used for problem solving as opposed to anxiety. So basically what we did in this experimental group or intervention group is that in that 25 minutes, first we said to students, physics department really wants to understand your concerns about being in this class. Why don't you write on this sheet of paper what your concerns are? Notice this is the first week of classes. Students wrote a lot. Finally, at the end of 10 minutes, they had to be told, stop, this is it. And after that, we showed them some testimonials from students in previous years who actually had similar concerns about being there. But then they worked hard, worked smart, took advantage of all of the resources, used deliberate strategies for learning, and went to the instructor's office hours. They went to the teaching assistant's office hour, worked with other students collaboratively. And lo and behold, what they found is that they did really well. So all of these students, some of the names of these students were men's names, some of them were women's names. And in their own ways, they were all saying the same thing, which is, I embraced my struggle. I realized that the key to doing well here was to work hard, work smart, and pat myself on my back. And then after all these testimonials, students were asked to get together in groups of 4 or 5 and discuss what they wrote on that sheet of paper at the beginning. And why don't students realize that struggling is common and that it is a good thing, because that's the only way to learn something new. Now, notice that students were asked to actually discuss why don't people realize. That means they had to buy this premise that struggling is indeed the stepping stone to learning something new. And and this kind of thing is common, which is actually true, right? So we really wanted to believe the truth. And so after students discussed this, we asked students to actually, report out what they had discussed. And lo and behold, all of the different groups pretty much discussed the same thing. Okay. So notice that this is normalizing adversity, and it's also helping them recognize that struggling is actually a good thing, because you will get over it if you embrace your struggle, use it as a stepping stone to learning something new, and you'll be on to the next thing and on to the next thing, on to the next thing. And of course, it is also our job as instructors to create the opportunity in which we create opportunity for productive struggles, right? We need to understand what is a student's initial knowledge and give them activities that they'll struggle with that will really stretch their learning. And then when the learning is stretched, then they'll be on to the next level and on to the next level. So the point is, we also have a job in terms of what kinds of tasks should we be giving to students to work on so that they will struggle. And if they struggle, they'll be proud of themselves and pat themselves on the back, right? So I'm not going to go through all of the things that, happened. If anybody is interested in the transcript, I'll actually show it to you because, okay, this is one of the testimonials from one of the students, Allison, who's an electrical engineering major. So this is actually a true testimonial from a student and we had similar testimonials from students who had really done well. And after that, we got students to talk about why don't people realize that other people are struggling too, and that it's a good thing to struggle because that's how you learn something. If that was the end of it, by the way, some women actually came at the end of the thing and they said, thank you so much for doing this. We could have just done problem solving in the entire recitation. We are so glad that you actually did this because it's making me feel so much better. Students were like, oh, are there other people in this dorm? Let's work together. So students actually, the way that they were interacting with other students after that was very different in those recitation classes compared to the control group. If that was the end of it, I would have said that's still good. But what we found is that when we look at the performance, for example, of men versus women, what we see is that in the social belonging group, there is no difference between men and women. The brown one here is women and blue is men. Whereas in the control group, women didn't do as well compared to men in terms of their course grade. The same thing happened when you look at, again, their total grade, the grade point that they got in physics. And again, what you see on the top is that, by the way, here the intervention group is the red thing and blue is the control group. And if you look here, this is men versus women. And you can see that there's no difference between men and women in the intervention group in terms of the grade point average out of 4. On the other hand, there is for the control group. Similarly, if you divide students by race based upon white and non-white students, same thing. You can see intervention group, there is very little difference, whereas in the control group, there is. The same kind of intervention was also done in biology at my university, and we found very similar results, for example, for white and non-white students. So you can see that a small intervention that creates a safe environment for students that tells them that they should be proud of struggling and they should be proud of failing because that is a stepping stone to learning something new, and in which you actually give to students productive tasks in which they'll struggle productively will go a long way in helping all of the students learn and feel safe in actually struggling and failing, because that's what we should be doing. Otherwise, the people who are actually underrepresented, the ones who are first generation college students, and if women are represented, those people are the ones who are not necessarily going to participate fully in any conversation because they'll be like, what if I say something and it's not right? What will people think? Those kinds of things should not be there. And you, as a teacher, you have the power to empower your students by telling them very explicitly the norms in your course and the fact that struggling is the key to learning new things. I also wanted to say that the same kind of thing is true if you're trying to mentor a student in any kind of research. Again, if you need to validate students, you need to affirm students, and you need to make it clear to them that everybody struggles in research. It's never going to go in a straight line. You go 2 steps forward, 2 steps back, two steps forward, 1 step back. If you're slowly drifting in a certain direction, that's already a good thing. Maybe in several months, you might not even make progress. Because if you don't actually tell them those kinds of things, then the people who actually will doubt themselves the most are the people who don't have role models, and they might think they are not making progress. They are not doing a good job, and they might even drop out. So if you really want to make sure that you exploit the talents of all of the students, you have to create an equitable and inclusive environment, even in your research lab, and make sure that all of those students actually take advantage of their struggle, and all of them feel that struggling is a good thing. So long as they are actually learning from all of the mistakes they are making, all the struggles that they are having, they are actually doing great. That is not failure. And so keep telling them those kinds of things. Otherwise, the people, as I said, the ones whose parents did not go to college, the ones who are from low income, the ones who are like, maybe women in disciplines especially in which they don't see a lot of professors who are like them, they are the ones who are more likely to question themselves. So I think that, this is also a good lesson in terms of how we should be mentoring students in our research as well. So I would like to stop here and take any questions. Thank you for your time. Did anybody have any questions for me? I see that Mansi has her hands up. Mansi, did you want to ask your question? Okay, maybe that's a mistake. Anybody has any questions?
Yes, ma'am. We have a question from Dr. Charu Dogra. So she's asked, how's heterogeneity of the class in terms of social economic backgrounds considered? It's a big variable in Indian classes. Will it not reflect the outcome?
What kind of variability not affect the heterogeneity in terms of social economic background? Okay, yeah, that's exactly what I'm talking about, actually. So I was talking about exactly the same thing. So what I'm saying is that, social and economic backgrounds can actually produce a lot of heterogeneity. And if we as instructors, that's exactly what I was saying to you, that if you actually do not account for those things, and if I were coming from a poor family, or if my parents never went to college and I was in your class, and if you never actually strengthened the defenses, and if you never actually said to me, said to the entire class that, no, everybody belongs here. Everybody actually will excel if they work hard, work smart, use the deliberate strategies, and do not actually worry about the fact that you are struggling. If you don't know something, nothing, you can never be wrong. Just, participate in discussions, feel proud of failing, because those are all great things. I think those students will otherwise not participate fully. And that is part of the reason we never actually really recognize the benefits of diversity, because the students who are actually not privileged, like those ones that you're talking about, the ones who come from low socioeconomic background, or the ones who came from families in which parents have not gone to college, those are otherwise not feeling like they belong. They don't see other people like them. And so we have a lot of power to empower especially those students and make them feel like they do belong there. And we have to particularly affirm them. We have to particularly validate them. And when they are making progress, we have to especially say, you are doing a good job, because they don't have, my kids, I know that they are going to do well regardless, right? But the point is, that is not true for a lot of people who are coming from those kinds of schools. So yes, those are the people that I'm telling you, you can empower if you think that you as a teacher, not just have a role to play in terms of strengthening students' defenses, but also strengthening their offenses. And when I call this intervention, that is a short intervention, notice it's done at the beginning of the course. The goal of this intervention is really to actually create a culture in the classroom. And I can actually send to either you, Princey, or somebody else, a clip from a faculty at Ohio State University. He says, it changed my life when I actually did this intervention, because, now in the US, many instructors have done this kind of thing. And seriously, they see that the culture in their class is different. When they actually think about doing these kinds of things at the beginning of the course, students start feeling proud of struggling. They start saying, yes, I'm having difficulty with this. Let's work together to figure this out. And notice that this is coming from you, right? That somebody that they look up to. So it really has a positive impact on them.
So very rightly said, it's our role to create equity, and we would, as you said, we would love to see that clip. And we don't have any other queries, but we have many appreciating messages for you, ma'am. So thank you for this enlightening talk, and your work is truly, truly inspiring. And I again thank you on the behalf of MICROSPHERE for patiently answering all the queries and taking out time at such an odd hour and addressing our participants. So thank you very much.
Not at all. I actually, not at all. In fact, I wanted to end with one anecdote, actually. So this is, there was this woman who is now an English professor at University of Michigan Ann Arbor. Her name is Eileen Pollock. She actually got a bachelor's degree in physics from Yale University. Okay. And she now is an English professor. So she actually wrote a book called The Only Woman in the Room, in which she talks about her experiences. And at Yale, they had to actually do a senior thesis. So when you are in your senior year, you have to actually write an undergraduate thesis. And she says that she did it on theoretical work. And when she actually solved the problem, she ran to the professor triumphantly because she was so confident that he'll tell her to go to grad school. He'll say, oh, Eileen, you've done such an awesome job. Why don't you think about grad school in physics? And that's exactly what she wanted to hear. But when she knocked on his door, he's like, okay. And she said that she just went from being triumphant about having solved this problem in her thesis to feeling like she was at the bottom of the ocean, right? Because here she's the only woman in this discipline. She doesn't know whether she's doing well. She doesn't feel like she belongs there, right? And here, when she thought she had done something great, her thesis advisor had no appreciation, nothing. He said nothing to her. And she said even in the book that, I felt like asking him, do you think that I would be good for grad school? Should I be going? But she said, if I had to ask, then I must not be good, because otherwise he would have said this to me himself. And so you can see, and the interesting thing is 30 years later, this is the first woman to major in physics. When she actually went back to Yale because she wanted to write her book because she still misses physics, she had breakfast with the same person, and this time he was the undergraduate advisor, and she asked him point blank, because she's much more mature, what did you think about my thesis? And he said, oh, Eileen, that was actually exceptional. And she said, really? Why didn't you tell me then? Do you ever praise anybody for doing a good job, or do you ever tell them to go to grad school? And he said, no, grad school is so hard. I don't praise anybody, because I don't want to give them wrong impression. Now, notice that the people that you will actually be driving out of a particular discipline in science will be the underrepresented people more likely than not, because those people don't have role models. They don't even know whether they're doing well. They need some validation from people who are teaching them. So please remember your role. You can really empower your students, make a big difference in their life. And so I basically, want to see that all of our students are excelling because of all the empowering that all of you are doing to the students. So thank you so much for having me, and have a great conference. Thank you.
Thank you so much, ma'am. Thank you for all the motivating words.
Thank you. Have a great conference. Thank you.
Thank you so much. Okay, so I would now request Dr. Gauri to introduce our next keynote speaker. Over to you, Dr. Gauri.
Thank you so much, Princey. So here we heard about the role models. So we again have a role model here. You know him by now. Our next speaker is Professor Rooplal. Professor Rooplal is the NASI Senior Scientist Platinum Jubilee Fellow at the Energy and Resource Institute, ISC Complex, New Delhi. He started his career as a lecturer in Sri Venkateswara College, University of Delhi, and has served for nearly 13 years and became Professor of Molecular Biology at the Department of Zoology, University of Delhi, from where he superannuated on September 30th, 2018. He has over 40 years long-standing experience in teaching, administration, and research. Professor Ruplal has specialized himself in the area of microbial diversity, genomics, and metagenomics. He has extensively explored the molecular and genetic aspects of SCH degradation, explored microbial diversity, sequenced more than 30 bacterial genomes, carried out about more than 5 comparative genomic projects, and in fact, 5 metagenomic analysis. Professor Ruplal was editor-in-chief of INJM, the Indian Journal of Microbiology, from year 2006 to 2013, and is also serving as a member of the editorial board in various leading scientific journals like M Systems, Environmental Microbiology, Environmental Microbiology Reports, BMC Biotechnology, BMC Biochemistry, Microbial Biotechnology, and many more. He has over 240 publications in peer-reviewed journals with 8,224 ISI citations and H-index of 46. Sir has delivered more than 300 lectures in the area of biotechnology, microbiology, genomics, and metagenomics at various national and international platforms. Sir is also recipient of various prestigious awards and fellowship. Today, I gladly invite Professor Ruplal for a one-to-one talk to all our students present here, along with the teachers and the faculty of various colleges. I welcome Professor Ruplal now to deliver a very insightful talk. Sir.
Right. So, are you able to hear me now?
Yes, sir, we are.
All right. Okay. So, it's really a lot of work that Shally has done, and her team has really brought out a very good combination of speakers and themes, and I'm very sure this probably conference or workshop at the end will give a very brilliant message to many of the students and colleagues who are participating. So thank you very much, Shally, for working so hard, and her colleagues for the past so many days uh, to bring out actually this nice workshop today with a combination of many good speakers. Now, I was listening to Dr. Chandralekha's lecture. I hope I can't see her, but I think she has already left. But when I was looking at what actually I did in my before I start actually letting you know the importance of microbiology. The students who were there with me, actually, I can now count actually out of 75, some 65 of them were women, actually. So also could be an exception, but only wanted to tell Chandralekha when she showed a slide that there was only one woman in the conference, Marie Curie, actually, which was held way back sometime. And there were all scientists were males in physics. But there were times in the lab when I was the only man in the lab, and there were some 10, 15 students who were women working in my lab. So when I look at it, I find another thing that has happened, of course, not probably was not really my intention, but empowerment of so many of my colleagues now who are taking up this issue of women empowerment and also microbial literacy, and at the same time they are also concerned about underprivileged students, and probably out of more than 700 or 800 registrations that we have got, I think many of these schools are government schools where probably the idea of again holding this workshop is to ignite these students at the very young age and making them aware of many things. And one of the issue, very rightly, that was raised by Chandralekha was women empowerment and also igniting students and especially underprivileged students. So today, actually, I'm especially speaking on the importance of microbiology in our day-to-day life. And probably this lecture I have delivered at number of places, and again repeating it. The idea of repeating this particular lecture at many places is to carry out a determined effort, and that is being done now by my colleagues. I can see actually some 20 or 24 of you have taken up this issue with a very, at a very different level, and now aggressively propagating microbiology, not only among the students or children, but and in India. But I'm very sure our these efforts will one day will become a sort of international on it will be heard on the international platform. So what I wanted to convey again to most of the students who are listening to my lecture for the first time, that there are a large number of microorganisms which we never noticed. And these microorganisms, which you call them as bacteria, fungi, viruses, protozoans, they are almost everywhere in all environments, be it hot springs or it could be a space or any other extreme environment. And it's only now, because of the coming up of newer techniques and tools, that we know that the number of microorganisms on this particular planet is something around 0.9 to 3.2 into 10 to the power 30 microbes. Now, if you look at the total mass of the living organisms on this planet, it comes to 30% of the total. So that means our 30% of the living biomass on Earth is occupied by microorganisms. And the number of viruses, that is something around 1 into 10 to the power 31 viruses on this particular planet. So with so many organisms, microorganisms are on. When I'm speaking to you, probably I'm interacting with not less than 30 million of microbes, including bacteria, archaea, fungi, and protozoans. So with so much number of microbes around me, can I actually live without knowing them? I think it is really very difficult if we do not know the role these microorganisms are playing. There was microbes, they are very important and abundant in our society, and we are not aware of their benefits and havoc they can cause. Probably Shelley mentioned, and everybody will mention, that a small virus which is around 70 to 90 nanometer. We call it as SARS-CoV-2, and also we call it as coronavirus. It has caused a havoc, actually, locked down the entire world. So it is one only single virus. And if you do some sort of calculations, you realize that the size of this virus is so small that you can, in fact, if you can calculate, that weight of one coronavirus is something around 8.5 into 10 to the power minus 21. And that is around 8.5 artograms. And worldwide cases, if at one point of time, there were around 2 into 10 to the power 16 million infected people. So the total mass of this particular virus that infected so many people was less than 1 gram. So the 1 gram or less than 1 gram of virus, with this infection, the entire planet is on its knees with just a gram of virus taking over. So you can, this again reflects the importance of bacteria, viruses, archaea, fungi, and protozoans and their association and benefit and harm they can do to the society. And you already have experienced it. So many people ask a question, why was the importance of microbiology not known earlier? Why did we only realize it now? It is basically because we had never been able to look at it. Microorganisms mean very small organisms, and not many people used to work with them. Number one. Number two, there were not many tools. Like, I will not get into very specific details, but like sequencing techniques and computational biology, that now actually brought into focus that we had never been able to even depict or culture 0.1% of the total microbes on this particular planet. So 99.9% of the bacteria, archaea, and viruses went unnoticed, and it is only because of the coming up of these neo tools that now we are able to know that there are so many bacteria or microorganisms around it. So this slide reflects that new tools are coming now because of the coming up of these new tools, a new discipline, which we call it as microbiome, community genomes, environmental metagenomics or metagenome, where you don't culture the organisms or don't culture the bacteria. We can simply isolate the DNA and then do some sequencing and computational biology and find out how many bacteria and how many organisms are total number of microbes, their genes, proteins, genomes, and what exactly they are doing. That is what we call it as a microbiome. So this is probably one message I want to convey, that we were unaware of the importance of microorganisms because we were never able to detect them to the extent to which we had been able to do it now. So this total, that's why now actually Shelley mentioned, I will come to that point later, the word microbiome day. Microbiome means community of microorganisms along with their gene and genomes and the functions they perform, being celebrated now on 27th of June every year. The purpose of this particular day is that we need to propagate microbiology not only among researchers and biologists, but take this particular message to the society and also to the children. So that's why this particular exercise has been done. And also, as Shelley mentioned, the International Microorganism Day, 17th September. It's again, actually, the mandate is to propagate microbiology among different, actually, components of the society, and also take it to that is the 17th September, Shelley mentioned, is International Microorganism Day, an opportunity to promote the diversity and variety of microorganisms, a way to encourage everyone to recognize and celebrate many ways microscopic organisms are important for human beings. And one of the projects that was initiated sometime in 2006 was Human Microbiome Project. Now, this Human Microbiome Project was mainly focused on knowing and understanding the role of microorganisms in human system. And surprisingly, when first phase of this particular project was completed in 2012, we came to know that when we are born, our genome is 100%. But immediately after birth, we actually get into our microorganisms, they get associated with us, and the number of genes and microbes that get associated with us is more than 90%. And also, surprisingly, we came to know that these bacteria, which are associated with human beings, they perform many functions which normally our own cell or genes cannot perform. And now we know, of course, this figure is varying, that the total human cells are something around 10 to power 30. But the microbial cells that get associated with us immediately after birth is something around 10 to power 14 and 10 to power 12 bacteriophages per gram of fecal matter are viruses. So much is the load of microorganisms on our body, their association, and 90% of the functions, directly or indirectly, are performed with bacteria that get associated with us immediately after birth, which was not known to us before the advent of newer sequencing and computational tools. So we are not our own cells. In fact, we interact with microbes, and our phenotype is a combination of all these organisms and all our cells and the genes coming from bacteria, archaea, and also coming from human beings that makes us to form a particular personality or a phenotype. Would like to mention that the maximum number of microorganisms per gram of fecal matter is something around 10 to power 11 to 12 actually in my colon. And that is what actually people have been saying gut feeling, which would mean that in fact, most of the functions, they are controlled in my body by the bacteria that are present in my gut. Not only that, these bacteria, they produce chemicals, and these chemicals actually, they are the signals which are carried to the brain. So this virtually actually, the human gut microbiome is also called as a second brain or virtual endocrine organ. And the composition of the second brain affects our first brain by a gut brain axis by producing large number of chemicals that are transmitted through nerves to the brain. So this human microbiome, we all now, everybody is convinced that it is in fact the human gut microbiome that is responsible for maintaining your health. And it, in fact, not to get into details, but to tell you that these are the gut bacteria that provide you immunity or immunomodulations. And any deviation from the normal gut uh, bacterial population can lead to large number of diseases. And now there is a lot of, we can search into the Google, and you will find that the microbiome is associated with any change from the normal microbiome. We call it as dysbiosis, and it can lead to large number of diseases: obesity, malnutrition, diabetes, asthma, liver diseases, heart attack, depression, blood pressure, arthritis, almost everything. The list is very long, but I have only listed some 13 of the diseases. If you look into literature, you'll find almost every disease is now getting associated with the bacterial population that is present in my gut. We also know today what are the bacteria which are beneficial and what are the bacteria which can cause, or they're becoming overpopulated in my gut or anywhere on my system, can cause large number of diseases. So the message which I wanted to carry, that yes, there are so many microorganisms around, and to maintain a good health, we need to understand that that we need to actually do something actually to maintain a good population of bacteria that can maintain us healthy. Will not get into the details, but we also published a paper where we actually studied the gut microbiota or gut microbiome of tuberculosis patients, those who were given antibiotics and those who were living with the tuberculosis patients. Our observations reflected that, in fact, in tuberculosis patients, there is an increase in the population of bacteria that produce short chain fatty acids and also propionate producers. And all these are responsible to impair the immunity. As I mentioned, that there are many bacteria around, not only in my gut, but when I go to actually ATM to withdraw actually some money, in fact, during that particular exercise, I get bacteria or swap bacteria. And in fact, it already was there in the newspaper. Then when you shake a hand, you exchange around 124 million of bacteria. And when you do high five, around 55 million bacteria. So this is what actually everywhere, wherever you go and interact with anything, you exchange bacteria. And now to change, or those who, there were large number of diseases, there are large number of diseases for which there is no cure. And now actually people are talking, and already this is being tried actually, fecal transplantation of healthy microbiome from the gut. So that is what is being done. And there are now probably, you will hear actually in subsequent lectures, that there are many probiotics, combination of large number of bacteria that are already coming up in the market. There are industries which are actually sending you the kits and in order to collect the samples from healthy individuals to be used later in order to cure large number of diseases. So that is the type of thing that is happening. And probably in 5 to 10 years time, you'll see a lot more diseases are being treated by bacteria, by bringing healthy bacteria into the gut or into the human system, rather depending upon antibiotics. So if we know what makes a microbiome function, what are the normal bacteria, what are the healthy bacteria, we can figure out how to keep ourselves healthy or fix a particular type of problem. Now, I will give you one example. I had given it a number of times. I was obese and my weight was something around 90 or more than 90 kg, around 94. And I read the first paper in 2012 from Human Microbiome, which appeared in Science, that in fact obesity and leanness is because of, not because of you or your own genes. Of course, they too matter, but it is basically because of the type of bacteria that reside in your body. And in order to maintain a healthy lifestyle, a proper or healthy bacteria in your gut, you can do three things: change your lifestyle, change your food habit, and do not take any drug or antibiotic. In order to do that, actually, I was determined in the year 2012. When I was obese around 97 degree, I realized and I read a paper that there are primarily two types of bacteria, firmicutes and bacteroides. Firmicutes are fat lovers, and the other class, other phylum, is carbohydrate lover. In lean people, carbohydrate lovers are more, and in fatty people, obese, those who are obese, the population of firmicute is more. You can change this population by, as I mentioned, by actually doing three things: lifestyle, food habits, and don't take any drug or antibiotics. So I started doing that. I changed my lifestyle, started doing exercises. In fact, walking a lot, almost every day, going to the gym, and then eating something which actually different from what I had been eating. I stopped taking fatty food. In fact, stopped to some extent taking chapatis and rice. And as a result of that, in few years' time, in 4 to 5 years' time, I reduced my weight by 20 kg. Now I'm 70 kg more healthy. And in fact, my blood pressure and all other parameters, they are so normal that I really feel very healthy and very young and do not get tired. So that is what to convince you, I gave. In fact, Chandalekha mentioned that if you can mention your own examples, they carry much more weightage to the audience, to the students. If you can tell your own struggle, probably they get better convinced. Yes, everybody actually faces such type of situations. It is not unusual with me or with a particular person. So if you are really giving your own personal example, probably give you a very convincing answer that yes, you can do this job. Because we have neglected this microbiology, we had been teaching actually A for apple and B for banana. We have started an exercise now. Why not to write books, and why not to teach children and school children A for Orthobacter, B for Bacillus, C for Corona. We have written large number of writing, in fact, and one of the editorials that appeared in environmental microbiology, you can read it: urgent need for microbial literacy in society. Then we are translating it in Hindi. Then also we have written another editorial for school children. And Shaili has already mentioned that we are taking up this particular issue, taking microbial literacy to the society and to the children by organizing large number of outreach programs. And probably the beginning of this particular program highlighted the number of such outreach programs that we have taken in order to excite young school children, tell them and let them know actually the importance of bacteria, and interacting with them, encouraging with them. We are now, the Shaili is leading this particular exercise along with our 20 or 24 colleagues, where we are writing a special issue, flip books. And this is appearing in Indian Journal of Microbiology, where flip books have been written. They are being translated into Hindi. So probably in a couple of months, you will know actually that when these books will be in the market. And not only this, we started some 20, 30. Now all my young colleagues, they have taken up this particular mission. Not only that from my team, but international scientists have also come into this particular group in order to propagate microbial literacy among society and children, and also make administrators aware of the role of microorganisms in our society. We are also organizing a very big conference where people are speaking on microbiome, microbiology, and role of that is the importance of microorganisms and the message that we want to convey to the children and to the society sometime in November 15 to 18. And if you really want to be a part of this, probably go to the website and see how exactly you can get into this particular thing. So with this small, I only wanted to ignite actually wonderful job that Shaili and all my 25 colleagues, they have done it. I'm very sure Shaili and other team members, the way you are taking up this particular mission, I'm very sure that one day actually this particular something which we have actually thought of will be materialized. And probably I would request through this particular presentation, as Chandralekha mentioned, that we also make this as along with this, we can also take up women empowerment and empower underprivileged students so that they become leaders in the society. And request everybody who was listening to my lecture, be our ambassadors, propagate microbiology in this society. Go to your home, tell your parents and tell your brothers, sisters, and almost friends that microbiology is very important. You need to understand the role of microorganisms, their association with us, in order to lead a healthy and prosperous life. Thank you very much.
Thank you so much, sir. Thank you very much. I feel that it is my honor that I have been asked to offer a vote of thanks on the occasion of International Microorganism Day to you, to Professor Ruplal, who has a long list of peer-reviewed research journals and tremendous experience on microorganisms. I think we have been fortunate to have a renowned identity like you in the field of microbiology today to celebrate this day with us. Like every time, today also I feel motivated and encouraged by your talk. Your simple ways of explaining microorganism has inspired me always, and I think all our participants also. And I must say, you have very well highlighted the need to spread the awareness about microorganisms, that it is not only limited to science or to a field of biology. We need to spread it awareness among all groups of society, and we need to cultivate this habit. I feel that the students are going to take up this to the next level. They're going to spread up this message, and sir has also very well explained and highlighted on the importance and all the aspects that he has been working on. So I request all the participants to encourage yourself, your classmates, and your family members about the importance of microbiology in our life. Moving on further, thank you very much, sir. Moving on further, I would like to invite Dr. Mansi Verma, who is an assistant professor at Sri Venkateshwara College from Delhi University, to invite the speaker for the next session. Thank you, everyone. Have a great day ahead.
Thank you, Jaya. Now, it's my pleasure to introduce the next speaker, Dr. Renu Solanki. Dr. Renu Solanki is currently teacher in charge and assistant professor in the Department of Zoology at Deen Dayal Upadhyay College. She has been working in this college since 2013. She completed her graduation from Daulat Ram College, University of Delhi in 2004, and MSc Zoology with cell biology specialization from Department of Zoology, University of Delhi in 2006. She completed her PhD from university in 2013. Her topic for research was isolation and characterization of actinomycetes and analysis of these their antimicrobial potentials. She has expertise in microbial technology, microbiology, and molecular phylogenetics. She has many research papers and articles in peer-reviewed national and international journals of repute to her credit. She has actively participated and presented research work in several national and international conferences, symposia, workshops, etc. With this, I would really like to invite Dr. Renu Solanki to kindly deliver her talk.
Thanks, Mansi, for the introduction. So let me first share my screen. Is my screen visible?
Yes. Yes, it is.
Okay, a very good morning to all. Myself, Dr. Renu Solanki, Assistant Professor, Department of Geology, Deen Dayal Upadhyay College, University of Delhi. The topic of my talk is the rise of microbes and microbiology. As we know, microbes are very small or tiny microorganisms. We can't see it with the naked eye. So for that, we need the microscope or the other instruments and the devices. And in the microbiology, we study about bacteria, viruses, fungi, algae, and protists. In my talk, I shall talk about the discovery and inventions in the field of microbiology, along with contributions of the scientists, because of which today we are able to see the microbes and understand their biology in a better way. So let's start with the So the first thing I would like to mention is the discovery of the animalcules, that the first animal-like microorganisms, and the who discovered it is the name of the scientist is the Anton van Leeuwenhoek. You all will be surprised to know that Anton van Leeuwenhoek had no school or the college education in which he had expertise in the microbiology. By profession, he was a merchant, he was a fabric designer, but his business was to observe the things very closely. So, for that, he developed around 500 microscopes, and this is one of the replica of his microscope. You can see it is a very simple device, it is a brass plate, and in the center is a lens, and in the front of the lens, there is a sample holder. So, with the lens, he observed many things. As Dr. Shelly Anand in the beginning of this conference has mentioned, that it is the he discovered the animalcules, that the first microorganisms, and on the 17th of the September, he wrote a letter to the Royal Society in which he mentioned about the animalcules. And to honor this person who has no background of the microbiology, we are celebrating the 17th September as International Microorganism Day. And this is the picture of the first bacteria which he wrote in the letter to the Royal Society. So you see that he is a merchant. He was a merchant. He was a fabric designer, but his keenness to observe the things led to the discovery of the first microorganism animals. After that, he observed many things like the human blood cells, spermatozoa, and many more. So, so the next thing is the discovery of the cell. Robert Hooke was a physicist. He gave the theory of many theory of the physics. Like he also observed the rotations of orientations and the rotations of the planets. But the Robert Hooke what contribute is contribution to the to the microbiology is the discovery of the cell. He was considered as an illustrator, which I will mention later on. First, I will tell you about how he discovered the cell. He also discovered telescopes. He also discovered the compound microscope. Under this compound microscope, he observed a slice of cork, and what he observed that there were some empty spaces. This is the actual picture which he observed under the microscope when he took the slice of the cork under the compound microscope, and he observed that there are some empty spaces. The cella word is derived from a Latin word which is the cella, which actual mean is the empty. So he observed, he gave these empty space as the name cell. Actually, he discovered the plant cells, and later on, the cell this became the theory of this became the basis of the cell theory. As I mentioned that he was an illustrator, because he observed so many things under the compound microscope that he revealed the reality of the unseen and the unobserved world to the people. So I would this is the book or Micrographia, in which his all the illustrations are published. He said this Micrographia to the Royal Society. This book is available online. I would suggest all of you to go through this book, that these are the for the first time he gave such a detailed pictures of the micro or of the many organisms, not not only the microorganisms, that he was considered as a very good illustrator, and people initially did not believe that this is the reality of the living world. So please go through this book. So now, to move ahead, I would like to mention the discovery of the first vaccine. This is the variola virus, which is the causative agent of the smallpox. In the ancient times, smallpox was a very deadly disease, and the situation was so worrisome that in the 18th century, around 4 million people were died in 18th century. 4 million people were dying annually, and not only the general public, but the five emperors or the kings belonging to the kingdoms were also they lost their life because of the smallpox. But at that time something interesting was happening, which was observed by the Edward Jenner, is that the dairy maid were somehow the immune, or they were the less affected by this deadly disease. So he also overheard that I had the cow pox and I need not to worry about the ugly scars which will occur on my face when I will get a smallpox. So the dairy maid, he overheard these statements, and it was also a belief at that time that the dairy maid were the immune to the smallpox. So he did an experiment in which he take the milk, he take the sorry he take the pus of the cowpox from an infected milkmaid who has contracted with the cowpox, and inoculated into an 8-year-old boy whose name was James Phipps. What he observed, that after some time that he did this experiment sometime in the May, and in July he repeated the experiment, but at this time he took the scabs from the smallpox infected patient and inoculated the James with the scab of the smallpox. But what he found, that the James was unaffected. There were the mild symptoms, and he was immunized to the smallpox. He was immunized to the smallpox, and the disease was not so deadly. So he started the vaccination of the people for the smallpox. But as today we know that the some of the people are have the apprehensions about the vaccinations. At that time also people were thinking that when they will be inoculated with the pus of the cowpox, there will be the growth of some cow like appendages on their limbs. But by the means of this vaccination drive, that is the situation of the smallpox could be controlled at that time. So in this way, the first vaccine, that is the vaccine of the smallpox, was discovered by the Edward Jenner. Now come to the discovery of the first antibiotic, and this is considered as an accident which changed the world. So the person who discovered the first antibiotic was the Alexander Fleming. In the World War II, he was deputed as an army captain, and he found that the soldiers were dying because of the fight also, but there were more mortality in the soldiers because of the infections of the some anaerobic microorganisms which are arising in their wounds. So he started working on the Staphylococcus aureus, and once time what happened, that he left a plate in his lab without keeping, and he did forgot to keep it in the incubator, and he went for the vacations. After coming back to the lab, when he saw the plate, what he observed that a mold was growing on the plate, and surprisingly, there were no bacterial colonies around these mold. So later on, he isolated the mold and identified it as the penicillium, which is a fungi. And what he found that he called it as mold juice. He concluded that it is not exactly the mold, but the mold is secreting some mold juice, that is the which is responsible for the no growth of the bacteria around its vicinity. So he later on this mold juice, along with the other two scientists, he was able to isolate the first antibiotic, that is the penicillin, and for this he was awarded the Nobel Prize in 1945. And this antibiotic was available for the soldiers of the World War II, which the agony or the pain was faced by the soldiers of the First World War. The Second World War soldiers need not to face the same problem of the infections because of the staph, because this penicillin antibiotic was very effective against a range of the microbes. So he said that I was not intended to discover the antibiotic. It is the nature who did. I only observed the what has happened. I did not ignore what has happened. So in this way, the first antibiotic, that is which is considered an accident which changed the world, and the first antibiotic was introduced for the welfare of the society, for the welfare of the human. And these are the plates which are available also on the online, that this is the penicillium colony, and there were the very less growth of the staphylococcus around it, just because it was secreting the penicillium. This is the pure culture of the penicillium notata. So the next is I would like to mention the Louis Pasteur, who is also known as the father of microbiology. And we all know today that we are able to transport milk, or we are able to milk. There is the milk industry is because of the pasteurization, a technique which was developed by the Louis Pasteur. What he showed that the heat killed the microbes, and the microbes will not be able to grow on their own until and unless there is a source of the microbe. So this is the swan flask, S-shaped flask, flask he used, and in this he showed, by means of this experiment, he also disproved the theory of the abiogenesis. Not only he gave the pasteurization experiment, he also developed the technique of the fermentation, many antibiotics against the rabies, anthrax, and all. And at that time, that the people used to approach him, and that there was the contamination in the wine and the milk, and he was approached to overcome the problem. So he introduced the technique of the pasteurization, on the basis of which the entire milk industry is proliferating. So this was all about. I would like to mention at the end that it is basically the contributions of the microbiologist. I can't say that these are microbiologists. Some of them are the chemists, some of them are the physicists, some of them are the common people. But the only major important thing about them is their observation power. So if you will keep on observing the microorganism around us, maybe you will be the next Alexander Fleming, or you will be the next Louis Pasteur. So, and the microbes are very important to understand, as in the subsequent talks you will discuss, they play an important role in the sustainable development. Also, there are the very many career perspectives in this field of the microbiology. Thank you all, and thanks to the organizers to give me an opportunity to speak at this platform of the International Microorganism Day. Thanks all.
Thank you, Dr. Renu Solanki. It was indeed really nice to see all the old pictures of like microscope invention, and even the experiments done by Alexander Fleming. With these words, I would now like to hand over this platform to Dr. Anjali Saxena from Bhaskaracharya College of Applied Sciences. Over to you, Dr. Anjali.
Thank you, Mansi. I now take the opportunity to welcome our next speaker, Dr. P.C. Ashwin Kumar. Dr. Kumar, currently Associate Dean Infrastructure, IIT Roorkee, is working as Assistant Professor in Department of Earthquake Engineering, Indian Institute of Technology, Roorkee, Uttarakhand. He graduated from Government Engineering College, Thrissur, Kerala in 2006 and completed his Master's in Structural Engineering from IIT Madras in 2010. He finished his PhD in 2017 from IIT Delhi and soon after joined IIT Roorkee in 2018. His main expertise area is with respect to design of steel structures and seismic retrofitting of existing buildings. He already has many publications in reputed journals and conferences and currently is guiding 6 PhD students and 4 MTech students. He has 3 years of structural design industry experience where he has awarded with Best Performer Award in the year 2012 by WSP Global Inc. India. He is currently the executive member in Indian Society of Earthquake Technology, meant for promoting and disseminating earthquake knowledge in our country, and he is part of CED 39 group of Bureau of Indian Standards assigned with the job of creating design guidelines for Indian design sector with regard to assessment of earthquake loads on structure. Today he will present a talk on microbes and sustainable housing. On behalf of our team, I welcome you, sir. I request all the participants to post their queries in the chat box and Dr. Kumar will address them at the end. Welcome, sir.
Thank you, Dr. Anjali. Can you hear me? And can you see my slide?
Yes, sir, we can.
Yeah, so good morning to everyone. Now I'll take you through another offbeat topic where you will learn how microbes are kind of contributing towards making sustainable infrastructure. So quickly getting into the topic, we know that we all live in certain houses, right? We live in RCC structures, reinforced cement concrete structures, our houses or industries or wherever we pass through, we see such buildings. And mostly in India, we use reinforced cement concrete to kind of make our structures. We'll get to what those are in terms of highlighting how microbes are kind of affecting or helping us in terms of getting to a sustainable solution. But before that, let's see how bad the current construction scenario is kind of affecting the environment. So as such, you know that carbon dioxide is part of that greenhouse gases, which we don't want to be emitted in large quantities. But sadly, the kind of materials that we use right now in our construction, which includes, like you can see, if I combine all the type of material that are used in construction, you can see, I'll switch on the pointer, you can see cement, ceramic, steel, lime, mortar, gravel are all the type of materials that are used in our construction. So cement, yes, you know what cement is and where it comes into picture. Ceramics is mostly your wall tiles, floor tiles and all. And steel is with respect to reinforcement bar, or it may be actually a structural steel like you see here, where there is no concrete, but only steel is coming into picture. Now, when I say percentage carbon dioxide emitted from common building materials, I'm not meaning that buildings and all breathe out of carbon dioxide or things like that. It is more or less what we say, the percentage, the total percentage is being calculated with respect to how from the time, the process with respect to production to its transportation and its execution at site, the amount of energy that is consumed to do all this process. And meanwhile, this particular release of carbon dioxide is associated with that being said, cement is the main contributor in terms of carbon dioxide being released into the environment, followed by ceramic and steel as the major contributors of the emission of carbon dioxide in our thing. So now what is the solution? Now, we have been seeing that there are not many new type of technologies being adopted in our country. Most common answer would be that yes, building a structure with concrete is low cost in comparison to going with steel. And if I tell you that these are the sustainable solutions that you have in front of you, nobody will say, no, I'll not build my home with that. But yes, some of these materials are being used, but widespread use of these materials are not used because They are not, I mean, you don't see them, you don't see their, what do you say, a kind of a long life kind of period. You have not seen them, so you don't believe it's property. But if you look at the kind of sustainable solutions that can be developed, or that can be researched, or some confidence can be brought into them, the materials include structural steel. Now, you may wonder that, yes, in the previous slide, steel has contributed about 18.7%. And why have I put it here? Is because structural steel is having a better kind of post-life kind of a scenario, in a sense that all our structures, in whichever structure you are living in, let's say our RCC structure is having a certain service life, which means we expect that structure to serve its functionality efficiently for a certain period of time. In our design industry, we say that an RCC structure, if you're living in a structure made of concrete and reinforcement bar, I'll come for students who don't understand concrete reinforcement bar. If you are currently living in such kind of structures, it is designed for a service period of 50 years, regardless of whatever type of load it may be subjected to. So in that 50-year service period life, the structure must not disintegrate or deteriorate while serving its functionality. Structural steel, after the particular service life, it can be 100% recycled. So that is the benefit that concrete RCC structure doesn't have, which we are currently using in India widespread, in a very widespread manner. That is not there for concrete structure, because we cannot 100% or 20-30% rebar can be kind of recycled, but concrete as such cannot be recycled. So it is a kind of burden of whatever energy that we have consumed, it is lying as a debris there. Same way, you can look at other solutions like rammed earth, straw, hempcrete, reclaimed wood, and even thermocol is currently being used as a structural member. Not, if you say a structural member, we'll come to what do you mean by a structural member and a non-structural member. Think of something like thermocol is being currently used to manufacture partition walls in your structure. So it is not bearing any load, but it is giving you privacy, it is giving you protections from heat. That is why we call it a non-structural member, because it is not bearing any load on it. Then you have ferroc. Then ferroc is like you start using the industrial waste to kind of make your platforms and all. Then cork is there. Bamboo in Northeast, where bamboo is growing at, the growth is there in terms of that particular material. People use it for building their homes. And precast structure, yes, it is made up of reinforced concrete. Yes, cement is kind of major component in there. And yes, cement is the one that is giving the maximum amount of carbon dioxide emission. So what is the benefit here is that all these precast structures are being made in a controlled environment inside a laboratory or a factory kind of a place where everything is done in a controlled way. So excess loss or whether it is excess use of cement or this thing or wastage of product is not there in that case. So that is why we have put it here. It is controversial, but still we have put it here. And then you have recycled plastic. Again, as a non-structural member, this particular component or material can be used for making partitions wall, flooring tiles, etc. Now, coming to our tiny contractors. When we say tiny contractors, as such the word when it comes into your mind, you know that yes, these are the people that may kind of make your home, kind of build your structure, have some kind of association with the structures that you've made here. So we have the three contenders with respect to bacteria, fungi, and algae. You can see that there are many names that you can see below them. And these are the names which I've pointed out have been explored in terms of how they can be utilized in making sustainable infrastructure. When we say sustainable infrastructure, these bacteria, fungi, and algae also come into picture in terms of giving an eco-friendly, a biodegradable material, not kind of having a burden on the environment. You're not worried about carbon dioxide. Actually, they are carbon negative in the sense that they consume that particular carbon and kind of make it a carbon negative kind of a structural system. So in all, this particular combo or individually bacteria, fungi, and algae can be used in making sustainable infrastructure. So talking about India, where we make concrete structures as the most, first we'll understand where the application, why is there a need for the requirement for bacteria, fungi, and algae to come in first place is to understand the process of making cement. So I'll take all the microbiologists on a journey with respect to how construction is being done in India with respect to reinforced concrete structure, first of all. So cement is made from the kind of components that are there in terms of making cement consists of calcium, silicon, iron, and aluminium. Calcium is mainly found in limestone, and all the other three, silicon, iron, and aluminium, is actually found in clay. So what we do, the first procedure is to excavate them from the site. Then the first procedure is to mix them properly. Mixing the process is there in terms of what is the proportion of each you want in them. And then after mixing them in correct proportions, you are kind of burning them or kind of producing heat. In first phase, it is provided in a tilted condition where the mixed sand, clay, limestone particles kind of pass through these hoppers and come into this inclined kiln, rotatory kiln, where initially due to the heat, the water is evaporated, and at the middle portion, the main component of cement, the compound which is created, which we call as a clinker, which consists of different compounds, which will come to dicalcium silicate, tricalcium silicate, tricalcium tetrafluoroaluminate, those kind of compounds form in them. The third process is to grind them. Before that, cooling is the thing. Then you grind them and then you package them. So you understand when we say carbon dioxide being emitted by the cement, when we say it all includes this kind of energy consumption that is coming into picture while producing this material, then storing this material, then transporting it, then kind of executing at site becomes a big problem in terms of the whole the whole process of carbon dioxide being emitted by cement. Now, coming to concrete, cement is just one part of the concrete. We are not coming into reinforced concrete right now. If you understand the whole process, you will understand why microbes are required in terms of helping out in making sustainable architect or infrastructure. Cement is just one component. You have just seen that cement contributes for maximum carbon dioxide, and how cement is made. A lot of energy is being consumed while making that product. When you talk about concrete, it is having four components. It's having water, fine aggregate, cement, and then coarse aggregate. Right. So what is the thing is that in a concrete, you mix them. All four component has very important role. If you have seen any construction activity going on in your surroundings or even at your house, all students, if you have seen them, what the person, the mason that is working would be mixing all these components together in a different way. If I say with respect to the cost of the concrete, cement is the most costly item there. But why, when we say that in India, it is a low-cost building material, why? Because we use bare minimum amount of cement, because coarse aggregate nearly captures around 70 to 80% volume of the concrete that we are making. Inside the pores, inside the coarse aggregate, whatever pores or holes or voids that are present inside the coarse aggregate, that is being filled by fine aggregate. Whatever pores are there inside that is being filled by cement and water. So what in all, it is about filling, compacting, getting a compact mass is what is required in terms of getting a good concrete. Now you will say water and cement. Actually, the main binding material, acting like a glue or a paste or something which is binding these fine aggregate and coarse aggregate, that work is done by cement and water together. So water, if you add more, if you see a mason without measuring beaker or having no idea about how much water is to be added, you should stop him. That is the main mistake which concerns with everyone sitting right now and listening to my talk, has to look around their house and see whether do you have cracks in your house, whether do you have to give money in terms of maintenance of your structure. ऐसा देना पड़ रहा है आपको, तो you have to think that you made a mistake while going with respect to a low-cost housing. When you went in for a low-cost person, a mason, when you gave him a small amount of money, what he did would have been, without calculation or anything, he would have added water to the cement in large quantity or very less quantity, and that may have caused the problem. If you had water in a very large quantity, क्या होगा, जैसे हम अगर पानी डालते हैं, एवरीथिंग सीमेंट एंड वाटर विल सेटल डाउन, ऊपर आपका सिर्फ कोर्स एग्रीगेट एंड फाइन एग्रीगेट रह जाएगा और वो कंप्लीटली बाइंडेड नहीं रहेगा. सो व्हेन लोड इज कमिंग, स्ट्रक्चर के ऊपर से जब लोड आएगा, your material won't have enough strength to sustain that particular load. So that particular ratio has to be maintained properly. If you go very low water, what will happen is that you can't work with that concrete mix that you have prepared. You will mix everything. You will use very less amount of water. It will once again cause pore size to be higher in that. But other than that, it will be very difficult for the mason कि आप column कैसे cast करते हैं. आप पहले form work लगाते हैं लकड़ी का. जो भी, that is one advantage of concrete is that आप mold in a sense कि आप कोई भी shape उसे दे सकते हैं. कोई भी आप एक cabin बना लीजिए, उसके अंदर pour कर दीजिए. Whatever size you get, वो जो भी आपको size चाहिए वो आपको मिल जाएगा. So water cement ratio plays a major role in terms of the kind of different reaction that takes place. Now, as I said earlier, clinker, clinker जो यहाँ पे form होता है, it is consisting of different compounds. So this is cement compound that you're talking about. And these compounds, tricalcium silicate, dicalcium silicate, these two are responsible for the creation of strength in concrete. And it only creates strength. ऐसा अकेले में cement बैठ के strength नहीं create करेगा. It needs water to actually act with it. That process is known as hydration. You must have seen that after a particular concrete specimen has been casted, आप उसको cure करते हैं. You add water, you water it for 7 days so that water doesn't kind of replete. So if you say that नहीं concrete को जितना जल्दी सूखेगा, when it becomes dry, you say that maximum strength आएगा, you are actually wrong. The more wet the concrete is at the end of the construction practice, you keep on wetting it. Curing is the process that is said. The hydration, which is happening as the reaction between the water and these compounds, actually provides more strength to it. ऐसा नहीं है कि concrete को overnight it kind of creates strength in it. It takes a lot of time. It takes years to get that concrete to reach its maximum strength. But हम as an engineer, what we say is, कास्ट करने के कुछ घंटों के बाद ही you kind of remove that formwork. It gains a particular strength. And as engineers, we measure 28 days strength as the average amount where the concrete would have achieved more than 80% of its capacity, total capacity. बाकी जो 20% है, it will take around some years to kind of get that particular strength. So if tomorrow onwards, if you're seeing some construction in your house or anything, ask him, do you know what is the water cement ratio? And ask him what is the gradation of coarse aggregate and fine aggregate. आप Badarpur sand, you must be hearing these kind of river sand being excavated. So concrete बनाने के लिए people are excavating sand which is from the riverbed. वो उसको what do you say in terms of environmental action? It is a wrong thing to do. You are deepening that soil deposit. So what is the reason is that why do they go for river sand is that proper gradation is there. You need a certain gradation. You need big rocks, small rocks, very small rocks, sand, dust, उस हिसाब से, so that you can fill in the pores. If you use one type of gradation of sand, it won't work. So it is a kind of symphony between all these kind of mix between all these materials which kind of play in terms of getting a good concrete. Now how do we engineers assess it? Is with respect to casting a concrete cube. इन सबका mix बनाया with the proper water cement ratio, or if you want to check what is the water cement ratio, हम इन्हें mold में डालते हैं इस size के proper size. Everything what I have said with respect to getting this particular cement कौन सा grade है, क्या strength है, already is given in particular Bureau of Indian Standards. Do masons follow that? No. Do you get a good structure? You might not know for next 20 years. After that, when the problem starts, you will then realize that something should have been done at the very beginning itself. So how do we measure the strength? We compress them. So the good thing about concrete is that concrete is good in compression, but very weak in tension. And the code also says that what is the proportion of water, cement, coarse aggregate, fine aggregate that we have to use to gain a particular strength. आज के date पे, if you are going ahead with respect to constructing a structure, you are required to be reaching more than M20 grade. M20 का मतलब है, M का मतलब है mix, 20 is the compressive strength at the end of 28 days. So you have to achieve M20, 20 MPa, Newton per mm square, at the end of 28 days in a particular building that is being constructed by you today. That is what the code says right now. For that, you have to go with cement, sand and coarse aggregate का ratio इस proportion में होना चाहिए, तभी you will get M20. If you go with 1:1:2, cement, sand and coarse aggregate in this ratio with a particular water cement ratio, you will achieve M25. If that person adds extra water or anything, that will cause early deterioration of your structure. Now coming to reinforced concrete, why we are doing all this, you will get to know in the next slide itself. So for the students' purpose, yes, what do we know as a structure that we live in is that कई सारे components होते हैं हमारे structure के, starting from the slab. There is a load path. I mean, आप एक structure को देखते हैं 2-story, 3-story, 4-story kind of a structure. You have to understand the structural configuration. आपके loads जो हैं, आप live load माने जाते हैं. आपके furniture, tiles and all are part of the dead load which is acting on the structure, sustained dead load which does not move around. That is why you are called a live load. If you place a dead load or a live load on a particular structure, the load path, the way the load flows is that slab से beam के ऊपर जाएगा, beam are the horizontal member. You can look around your house to identify these structures. You might be living in an RCC structure, but India में even brick masonry के structures भी बनाए जाते हैं, on top of which slab is kept, RCC slab. You can identify that, yes, the load from the top, अगर slab यहाँ पे है तो slab से beam की तरफ, which is at the periphery, which is provided here. बीम की तरफ फ्लो करता है. From beam it flows to the columns, and from columns it goes to the foundation. Now, as we said, concrete is in good in compression but weak in tension, which means अगर एक beam bend करता है, तो you can see that the top face of the beam is coming under compression where concrete is good, but at the bottom side it is good in tension is coming, so there the concrete has started cracking. And where only reinforcement bar, जो हम सरिया देते हैं, जिसको हम reinforcement bar, सरिया, which you see with respect to structural steel, or reinforcement bar जो steel से बनता है of different grade, that is what we use. So the load coming from the slab moves on to the beam, from beam to the column, and column to the foundation. And with proper mix being provided, जो हमने पिछले इसमें देखा कि proper concrete का mix होना चाहिए, जिसके basis में structure बनता है, then it is possible that even then what I'm saying is that concrete at the top portion will be fine, but again cracking is happening here. So when cracking happens, what will happen? The reinforcement bar will be exposed to external environment, which will be like water ingress करना शुरू करेगा, पानी अंदर आना शुरू करेगा. There are other reasons also why cracking can happen. It is not just that load is the enough. There are other causes like freeze and thaw. ठंडी में compress होना concrete का, गर्मी में expand होना, cracks will develop. Cracks are meant to happen, micro cracks. We are not talking about big, huge cracks that can happen. We are talking about small micro cracks even that can happen, which can cause the water to ingress inside the concrete. If that happens, if you see here, this is a building which is there in Delhi, which was recently surveyed by us, where you can see that the reinforcement bar is totally corroded. Which means एक अच्छा design engineer and an अच्छा contractor or a mason if you hire, and if you tell them कि भैया design this properly, he must have designed and provided a particular reinforcement bar dia in your structure. He said 20 mm का diameter का reinforcement steel bar दे दो alongside with an M25 concrete mix. Right now you understand what I'm saying. Then you will get a particular strength. But what happens? Duration. I mean, you have a service life of 50 years. You may be living in a coastal area where salt solution also comes in, which aids again for corrosion. Then if you're living in a city like Delhi, population also leads to carbonation, corrosion kind of an event. So there are different aspects with respect to alkali aggregate. Saying like this thing is that when you say cement and water reacting, we never mention the aggregates reacting because these are inert materials. Chemically active nahi hai wo. But sometimes when these people pick these stones or rocks from some arbitrary locations, they don't grade it properly, you might end up with having aggregates which are chemically reactive. That will provide expansive forces on the concrete. Crack develop hoga, water will increase and aapka strength jayega. You can see the rebar getting detached from the concrete itself. Agar aapka bottom side mein tension lene ke liye reinforcement nahi hai, concrete cannot take tension at all. तो आपका structure collapse हो सकता है, and that way many buildings are being brought down in Delhi and different places. You have to either spend almost entirely same amount to retrofit the structure or bring it down. Now coming to the main part, where our microbes can come in handy is because these microbes that I have discussed in terms of bacteria, you can see bacteria here and you can see the algae here. These have two properties. These have the same property in the sense that they can generate insoluble calcium carbonate. कि वो एक अगर वो microbe को surface पे, जैसे first way of protecting the concrete surface is biodeposition. किसी भी concrete surface पे आप इसका microbes का जो ये bacteria जो मैंने पहले contenders दिखाए थे, list of bacteria that have been worked upon, with nutrients, some calcium source उसके साथ, nutrients के साथ अगर हम spray कर देते हैं concrete surface के ऊपर, these bacteria with these nutrients can produce insoluble calcium carbonate. वो precipitate करते हैं and it provides a kind of what do you say उन pores को या surfaces पे अगर cracks है तो उसको बंद कर देंगे. So what will it do? It will not allow oxygen to enter. It will not allow water to enter inside that particular crack and cause further corrosion or deterioration of the structure. And that way, so हम microbes का first role क्या देख रहे हैं? This is for enhancement. कि if right now many of you who have bought a new home, living in a new home, you might be seeing at the corner of your eye that at the one particular location you'll find certain corrosion of structures happening, deterioration हो रहा है. 10 साल के अंदर ही concrete fall off हो रहा है. Falling off is just falling off from the surface. कि आपका beam या concrete member से गिर रहा है. Which means that कि जिस service life के लिए आपका structure design किया गया था, it is not attaining that. it is not attaining that much which has to be achieved. So what option we have? We need a low cost option in terms of enhancing the service life. So one way is enhancing the service life using bacteria and fungi, algae, in terms of their characteristics, that they can produce certain types of crystallite material or gel type of material which can block the ingress of water agar hota hai. So one way of doing that is biodeposition, surface पे spray करो. या दूसरा, जैसे हम आजकल करते हैं कि ग्राउटिंग करते हैं, हम प्रेशराइज्ड वे में इंजेक्ट करते हैं. वी आर नॉट यूजिंग एनी सिरिंज नीडल्स और एनीथिंग, बस एक रिप्रेजेंटेशन के लिए दिखाया गया है. यू यूज दैट सिरिंज नीडल या एक काइंड ऑफ प्रेशराइज्ड वे में आप इन क्रैक्स में यू कैन ड्रिल होल्स इन कंक्रीट एट डिफरेंट लोकेशंस, and then under a pressurized condition you can put in there. So when you put this microbes, nutrients and calcium source के हिसाब से which the bacteria uses to create insoluble calcium carbonate, वो उन pores को seal कर देगा, which means आपके rebar को थोड़ा और life मिलेगा, आपके concrete को और थोड़ा life मिलेगा. How much? It has to be explored. Now finally, अगर हम बाद में ये सब कुछ जो पहले biodeposition and manual injection is it's not prevention, it is like कि हम हो गया incident and then we are trying to repair it. We can do another way in terms of self-healing concrete के case में this has picked up pace in terms of microbes being used as self-healing, is that inert microbes in terms of कि microbes जो सोए पड़े रहते हैं अपने जगह पे, you are encapsulating when the concrete जब mixing होता है, आपने ये ड्रम बैरल ड्रम जो है या concrete mixer है वो देखा होगा, उसमें जब आप concrete mix बना रहे with cement, sand, coarse aggregate and fine aggregate, you add these microbes along with nutrient sources, calcium, to this particular thing. These bacteria will sleep for a long time. It has been seen that they can sleep up to 200 years also without getting affected. So if and all crack happens, which will happen eventually, मैं ये नहीं कह रहा कि ऐसा कोई structure reinforced concrete का है which cannot crack. Cracks will happen, water will ingress, and then what will happen is that the water, when it gets inside that particular concrete, it will kind of try to degrade the structure. तब क्या होगा? These microbes which were inert or kind of hibernating inside the concrete will get activated, and then they will start producing this calcium carbonate, insoluble calcium carbonate, which will again stop. So जहां जहां जरूरत है वहां वहां ये activate होते जाएंगे. That is why the name self-healing has come. So this is enhancement of that 50-year service life जो हमने बोल रखा था. Now another way is for creation of a new component. This is where all students who are hearing, every college student, every faculty who is hearing, can think of that some idea from here can be an idea for ma'am Helianthus ma'am. Can you please mute yourself, ma'am?
तेल लगाने से ठीक हो जाएंगे. वैसे तो अभी तक नहीं दिख रहे, लेकिन
maybe the organizer can mute them. Yeah, I'll continue. So all those students, teachers, and everyone who are listening can think about this particular idea in terms of creating their own company, in terms of an incubation company or some sort of this thing that we are talking about fungi here, specifically fungi in terms of mushroom. If you have seen mushroom growing in a forest area or something, you can see a fibrous white networks which you see here in this figure, which can grow. ये grow करता है in insurmountable amounts in terms of in widespread amount में ये grow करता है. And what you see here is kind of stubble or something like lignin जो दे सकता है उस type का material. You keep it as the base material, on top of which you add the nutrients, as well as you keep this particular mushroom को आप उसमें grow करने देते हैं. Within 15 to 20 days, that is one issue in terms of getting a product. You will see that this mycelium has grown all over the entire content, which is the thing here. So that acts in a way like this fungus, this mycelium which is growing, acts as a binder element, like cement की तरह वो एक binder element की तरह act करना शुरू कर देगा, which will give you an end product which you can use it for multiple purposes. People have started using bricks from them. People have started making partition walls from them. People have made furnitures from them. There is also a big building that you see here, which was made for a particular exposition or conference के लिए ये बनाया गया था, just to show that this particular material has a versatile kind of a use. So how is it helping is that कि जहां अभी आपके घर में आपका partition wall brick से बना होगा, correct? So as an earthquake engineer, why am I looking at this particular microbes and all is in a sense that earthquake is not a force which you can see. Wind की तरह या vehicle आके building पे टकरा रहा है या wind act कर रहा है. Wind as a pressure, you know that is acting on the building. You can measure it with wind vane, etc., etc. Earthquake is like earth is shaking. Earth is kind of quaking, or the way you can say is earth is kind of providing vibration to the structure. So what you see is that earth to shake kar raha hai. Why should my building be concerned about this? The problem is, think in the same way as you thought about while talking about inertia in class. आपके टीचर्स ने आपको बताया होगा, आप बस पे चढ़ते हैं, बस फॉरवर्ड की तरफ एक्सेलरेट करता है, आपका बॉडी पीछे की तरफ जाता है. तो you can think that inertial force is acting on the body. So if you consider that, Newton's second law, सबको आप मिलाते हैं तो buildings generate forces during an earthquake due to that inertial property. Mass ज्यादा है, ज्यादा forces act करेगा building के ऊपर. So if you look around you right now, if you see a brick wall that is being made, आपका partition wall most probably will be made up of bricks laid one on top of the other with again mortar, cement mortar उसके बीच में डाला गया है. Brick बनाना ही एक अलग problem है. You have seen brickins using this thing. So bricks का अपना weight होता है. But if you make a brick out of mycelium or things like that, it will be a lightweight structure. Lightness in weight of structure will relate to less amount of force being acting on the structure during an earthquake. So that is the advantage. Less amount of inertia. Rest of the members, beam, column, element, let it be there. That can be explored further. But it has to be seen in a way that how can we kind of save our structure? And it has been seen. I mean, I'm just adding upon and saying that when you say mycelium being put into kind of stubbles या crop का जो waste रहता है, जो आजकल दिल्ली में problem आता है कि winter के आते ही stubble burning शुरू हो जाता है, the stubbles के साथ mycelium का combination is found to be very good in the sense that you may think that ये stubble लेके करता है तो फिर तो वो fire protection उसका नहीं होगा, but actually after mycelium kind of engulfs it, the kind of property that it shows with respect to water absorption and etc., with respect to fire protection or fire safety ke basis pe, which is looked up as if in terms of how much hours of fire protection can the material give, is actually very, very good in terms of comparable to what concrete can give. So lightweight, thermally insulated, fire protected kind of a material when you have in front of you. Why are we using bricks? Again, jeopardizing our kind of structures. So what I have shown here is the previous contenders which I have shown, they have been researched upon. It is not like it is a new concept or something. It has just started. It is in its nascent stage, but things are developing. So in terms of that, we can see that the what is the improvement is that you can see if you add this to the concrete, there is an improvement in the compressive as well as the tensile strength. Even though, yes, we are still relying upon reinforcement bar, we cannot replace reinforcement bar because बहुत ताकत लगती है. But increase in compressive strength, it's not kind of deteriorating. Adding microbes to the structure is not deteriorating its strength. And it is reduction, if you add this particular thing due to the formation of insoluble calcium carbonate, the porosity and water absorption. Water absorption हम कैसे करता है? Cube बनाता है, पानी के टंकी में डाल देता है, certain hours. It is all based on scientific calculation, कितने घंटे डालने हैं, उसके बाद increase in weight को हम कैसे calculate करेंगे. In that way we say water absorption में कमी हुई है, which means water is not percolating inside the concrete. It is not taking. But my last slide is with respect to this. What are the problems? It sounds wonderful in terms of the new material that we are talking about, but the issue is scalability. अभी तक तो हम छोटे-छोटे bricks, छोटे चीजों में कर रहे हैं. It has been also seen that अगर आप इस bacterial इसको soil में डालते हैं, due to this particular deposition, the soil property is getting enhanced in terms of कि earthquake के लिए भी आपको मदद आएगा. अगर आपका vibrations are getting reduced even at the soil level itself, that is also helping me. But आप समझ रहे हैं ना कि एक building के चारों तरफ के कितने soil को, कितने huge amount of soil को मुझे वो ये करना पड़ेगा. That has to be seen. If you apply it for a big building, a big partition wall in real case, in the long term वो कैसे behave करता है उसको भी देखना पड़ेगा. Again, time and cost, आपका nutrients का cost. With respect to bacterial, इसका जो time लगता है 16 days for a mycelium brick to form, it is a huge time. आज के date पे bricks अगर बनते हैं, it can be given in a day or two. But mycelium bricks, if it comes in 16 days or 20 days for the exact material you want at hand, it has to be explored that how can we increase the time in which we can give it. Then जो nitrates और ammonia जो byproducts होते हैं, इसके अलावा भी in the whole process what is being created, that has to be taken care of if it is being created in very large amount. And the second last thing is with respect to bacteria, nutrients and injection rate. कि एक पर्टिकुलर यूज के लिए मैं कितना अमाउंट ऑफ बैक्टीरिया डालूं, कितना न्यूट्रिएंट्स डालूं उसके लिए, and at what rate and what depth I should kind of inject it into the material. This magic mix proportion has to be developed for each material, each application, each bacteria and etc. And longevity, this is the bigger question, that since it is at its nascent stage, जहां तक आपके existing concrete structures हैं, you have to take care about those structures. That is where enhancement comes in. लोगों का भरोसा आना चाहिए concrete से हटके to this particular thing. You need time. Longevity उस तरीके से भी मैं बोल रहा हूँ कि yes, for the structures that are already existing, to increase their life, उसकी लाइफ बढ़ाने के लिए माइक्रोब्स को यूज करना है. जो हम नए टेक्निक्स यूज करते हैं, नए टेक्नोलॉजी, उसका longevity भी हमें देखना है कि on a particular day, right after making that element, it might be looking good, it might be performing good, its material and mechanical characteristics will be very good, but बाद में क्या, after 20-30 years, what will be its characteristics? वो करके, develop करके, you should build confidence into the people so that you can bring in your company, your vision, your idea into the market as a passion in terms of कि आप biologist हैं, आप microbiologist हैं, you are a structural engineer, you want to do good for the society, you want to bring a sustainable infrastructure to the society. These roadblocks have to be kind of solved before you reach a particular solution. So I urge everyone to kind of contribute in that manner in terms of getting to a solution for getting into sustainable infrastructure. And with that, thank you, everyone.
Thank you, Dr. Kumar, for your enlightening talk. Your work is very interesting and different. The microbiology with construction work along with the tiny contractors is really appealing and has charmed all of us today. On behalf of our team, I extend a hearty vote of thanks to Dr. Kumar, who spared a tight schedule to grace the occasion. Thank you, sir.
Thank you. Thank you to all the organizers, and thank you, Dr. Anjali, for this opportunity. Thank you very much.
Thank you so much, sir. I now invite Dr. Jaswinder, Assistant Professor, Gargi College, to welcome our next speaker.
Hello, I hope I'm audible.
Yes, Jaswinder.
Thank you. So our next speaker of the day is Dr. Amjad Hussain. He's been very patient with us. Thank you, sir, and apologies for the delay. Dr. Amjad Hussain is a PhD in biotechnology from International Center for Genetic Engineering and Biotechnology, ICGEB, New Delhi, and a postdoctoral scientist from Harvard University. At Harvard, he worked in the area of bioengineering and cancer research and was also an active member of Harvard Innovation Lab. His work at Harvard led to collaboration with companies like Eli Lilly in the USA. He successfully commercialized the high-throughput drug screening platform through a startup company in Boston. Currently, Dr. Hussain is the CEO of Innovation and Incubation Center for Entrepreneurship and Principal Scientist at ISSER Bhopal. He is also the coordinator of Technology Development Center of the Institute. He is also the co-founder at Kenefis Therapeutics, a startup company in the area of CAR therapy for cancer treatments. Apart from that, he also does consultation for many biotech and edutech startups in India and abroad. Dr. Hussain is a TEDx speaker and is passionate about entrepreneurship, traveling, and teaching. I welcome Dr. Hussain on the virtual dais and speak about technology entrepreneurship. Sir.
Thank you so much, Dr. Jaswinder Kaur, and thank you, organizers, for inviting me for this opportunity today. Let me start sharing my screen. Can you see my screen?
Yes.
And should I maybe made it on the slide mode? I hope it is visible.
Yes, sir. Perfect.
Okay, great. So I believe that I've been given 25 minutes for this talk, so I'll try to finish on time, provided that there are not many questions. But we can take the Q&A at the end of the talk or to the chat box as the organizer suggests. So thank you once again. I'll be speaking more about science and technology entrepreneurship. My talk will not be heavily loaded with scientific data, but instead it would be more of a guiding sort of presentation for students who would like to start the company, who have a raw idea or a prototype, and who are a science or technology students, and they're looking for opportunities to start a company. So I will give some of the examples, some of the success stories, the companies which have become successful, especially they came out from the science background, from the academic institution, and how these companies have become very successful companies across the globe. And I'll also then give some idea on the procedures of incubation of a company, and then how to sort of raise funds and move forward. So, as Dr. Kaur mentioned, that I'm the Chief Executive Officer at Innovation and Incubation Center for Entrepreneurship at IISER Bhopal, and also a principal scientist here. Just to give you the disclosure, most of my training has been in Boston at Harvard Medical School as a postdoc, and also I studied a management program at Harvard Business School, has been associated with a couple of companies. Life Cell Technology was one of the company we started, it got acquired, and then Cellworks and Clone Lily, these are the companies which I've been associated with in different capacities at different point of time. So in my presentation, I'll talk about that who is an entrepreneur, what are the success stories, does scientist-driven entrepreneurship really exist? What is the incubation support system for student entrepreneurs? I'll give you IISER Bhopal example, but of course, there are very good incubators at IITs and across the nation in different national and regional institutes. And what is the right time to start a company? Do you think that you are a student and you are so young, or you it is not the right time to start company? So should you wait, or should you start right away? So that is something which I would like to begin with. So just to give you the perspective, if you see all these logos, these are not related to microbiology, of course, but these are some of the companies which impact your day-to-day life. And I wanted to convey this message to all of you, especially to those who think that entrepreneurs are greedy people, or they are looking to mint money, to make money. The fact is that entrepreneurs are real problem solvers. If you get up in the morning at 6 or 7 a.m., using the toothbrush, and then to start your day with, and then eating Kellogg's at the breakfast table, taking Uber or Ola, ordering stuff from Amazon, checking your profiles on LinkedIn, tweeting in the capacity, or staying at Airbnb, sipping a cup of coffee at Starbucks, or watching videos through Netflix, or shopping at Walmart, or maybe using a drug of a company like Novartis. Every part of your life is impacted by companies around you. And these are the ventures which were started by someone at some point of time with a bigger vision. And that's how they become successful. And of course, they are very capital oriented ventures, but they do solve problems. That's why you pay price for them, because they make your lives easy. These are some of the most influential people in the world, pretty much the richest in the world, in decreasing order, starting from Jeff Bezos. And they are the one who have started multi-billion, and maybe I would say a potential trillion-dollar companies. And they are very well educated. So there is a notion, or there was a notion, especially in India, that people think that people who are educated, they don't pursue business, but that is not the case. Out of these 15 people, I would tell you that 3 of them went to MIT, 5 of them went to Harvard and Stanford, and at different universities, and they started these multi-billion dollar corporations. Not only men, but there are very successful women who have also started these companies, and now they are the part of the billionaires club, and they are leading the companies globally, and they are making an impact. So the question is, since our audience are mostly science students, the question is that, do scientist-driven ventures really exist, or is it just a work done by MBAs or techies? Like can you also start a venture as a scientist, as a science student? And answer is yes, of course. If you see the picture, the gentleman is a Thomas Edison, and he himself, although he was one of the greatest inventor of his lifetime and even later, he himself started a company called General Electric, GE, and he's founder of multiple company. He owns so many patents, and there is a story about GE which I'm not narrating right now, but that's how it impacted economy of United States and how it generated employment and how it sort of changes the life moving from when people were when light was not even electricity was not even discovered earlier. So he had multiple patents, and then these are some of the modern age innovators as well as their academicians. Philip Sharp, who is from MIT, also a Nobel laureate known for the discovery of introns, have started multiple companies. Biogen, Alnylam are one of those. Robert Langer has more than 800 patents. At this point of time, professor at MIT, he has started more than 30 companies. And just to give you the idea, Moderna, I'm sure you're familiar with the name. Moderna was also started from his own lab. And right now, his own worth is more than $3 billion. So these are some of the very, very successful people who are doing cutting edge science, but also taking that science forward for the public usage, for the commercialization. So the products are available in the market. Their science is not restricted only in the boundaries of laboratory. Their science is not restricted only to the publication, but instead they're taking it out in form of products and services. I'll share with you another example of a discovery of a drug called Avastin, which is an inhibitor of VEGF. And this is one of the problem that I also worked upon when I was doing a postdoc at Harvard in Boston. The tumor which grow by increasing their blood supply. So there is one approach that people target or the drugs they target the tumor cells, but when tumor grow it require more blood supply for the nourishment of its own. And therefore another approach is that instead of targeting tumor, you can target the blood supply, you can target those vessels and using those inhibitors. And what we realized that this molecule called VEGF, which is called vascular endothelial growth factor, it was a cytokine which was discovered long back at Harvard, and they realized that this is the molecule which causes increased blood supply, which causes formation of new blood vessels towards the tumor. And this molecule was discovered by a gentleman, Hal Dvorak, who was also my postdoc mentor. Now he's retired. And then the company Pfizer, they discovered an inhibitor of this molecule called Avastin, which is still being used for the treatment of solid tumor. And the annual sales of this single drug of Pfizer is almost $2 billion, which is approximately 15,000 crores in Indian currency. This is another example that VEGF was also claimed by Genentech later by Napoleon Ferrara and Bob Langer, and then Genentech, they have of course a lot of drugs, not only the VEGF inhibitors, but a lot of other drugs related to VEGF and other pathways, and it was acquired by Roche for almost $50 billion, the company. So these are some of the examples to suggest that how these innovations, these discoveries from the lab have moved out of the lab and for the production and commercialization of these products while partnering with industries or licensing out those technologies to different industries. This is another example which I like to share with you using a personalized medicine approach. These are the type of companies which are coming up a lot in microbiology as well as in the field of cancer, where what we did when I came back to India from US, I was heading a research group in Bangalore. It was a cancer oncology company, and we had like a lab, like an academic setup, but it was a bigger lab. Almost 40 people were working in my group, and I was heading that group. And what we did, that we analyzed all tumor mutations, and we realized that these tumor mutations have some impact on cell division. And accordingly, we gave that score, we simulated that score, and accordingly we were looking that, okay, if these are the tumor mutations, then whether cell will proliferate more or less, which pathway will be more dominating or less dominating, and then accordingly which drug will work better. Because the problem is that, in most of the cases, if there are, let's say, 5 patients of breast cancer, you give them same drug, 3 may respond, 2 may not respond, because their genomic profile is different, their receptor profile is different. So we were trying to give a personalized touch to the treatment. Just to give you the perspective without going deep dive, that how we nailed it down and how we suggested that, okay, personalized medicine using genomic information can help in better treatment of cancer. And in fact, we worked with a lot of engineers from Google, and they also helped us creating the map of cell signaling pathways the way they created Google Map and GPS information. This was just a proposed simulation model that we used. So we categorized those genes into different category, like, okay, so these are the genes for cell proliferation, these are the genes for cell survival, these are the genes for cell death. And then we see which genes are more active, and accordingly we take a call that which drug will work and which won't work. So that was done using bioinformatics and validated using the wet lab approach. Now, coming to the microbiology, just to give you the idea about how microbiology driven startups are working, there are multiple gut microbiome startups are coming up. And this is one of the startup, day two, that actually talks about the food based on the gut microbiome. So what they suggest that, okay, if you have certain type of bacteria in your gut, this, of course, they will take the sample, and then accordingly, using a stool sample, they will tell you that, okay, this is your microbiome profile and you should use this food versus that food. And this is all automated. So, and then this is, I think, impacting lives of people, because a lot of people, they were getting obese. And you may have observed this, some of the people, if they drink even 5 bottles of Coca-Cola, they still don't get fat. Even if they eat a lot of non-vegetarian food, they still remain skinny. But some of them, they sort of, even if they eat one extra spoon of sugar, they start gaining weight. So partly that also depends on the gut microbiome. These are some of the new insights, and now people are taking these concepts forward in form of a company. And this company have raised more than almost 80 crores in funding. These are another examples of gut microbiome ventures. They are actually working on different aspects of it. For example, some of the companies are now coming up that, okay, if you have these kind of gut microbiome, A, B, C, D, E, F, then you have more chances of developing colon cancer. You have more chances of drug metabolism. You have better or lesser chances of delivering of the drug. Even one of the company, I'm not disclosing the name, in Germany, is working now on certain bacteria, that if you eat even sweeter thing, and if you are a diabetic person, then the sugar transport will not happen from gut to the towards the blood. Basically, even diabetic people can eat sweet stuff, because ultimately it will not be going to the blood and it will not impact the blood sugar level. It will, you like, a person can eat and it can get out in form of stools. So that is these are some of the new concepts coming up in the gut microbiome space. Just to give you some more examples, that these are some of the companies which are started by scientists, and also I would say they are very, very heavily science driven. I've already discussed a few, and for example, eGenesis is one of the company which is making humanized mice and humanized organ using animals. Foundation Medicine work on the genomic principles. They take the cancer samples and then do genomic analysis and provide you the right treatment. Joule Energy was started by an Indian person, Indian very, very rich, the owner of Microsoft, Sun Microsystem, Vinod Khosla. And then Calico is another venture recently got started in California. They are working on better lifestyle and life extension. Now you may ask this question that these are the examples in West. Do we have people in India who are core scientists and successfully started the companies? And answer is yes. There are many, but I'll just quote in interest of time only one. Yusuf Hamid is a chemist and entrepreneur, a PhD from Cambridge University, who has started the company Cipla. I'm sure we all know Cipla for the quality and affordable drugs that they provide to the Indian people, and also help in societal development while making the cost affordable. Now the question is that as a science student, what should you do to become an entrepreneur? uh, do you need to be an smartest person or a topper of a class? Probably not. Basically, for entrepreneurship, a student or a person should have, of course, the basic understanding of science, and if there is a good idea, but also an insight for commercialization. You should have an insight that, okay, how would you make this idea into prototype and then your product? How do you make your lab research into something which is sellable product, or at least close to that? And then most of us, what we have just the idea, or we have just untested hypothesis. So you also need to plan a little bit more so that you can test it a little bit. And at least if you have 10 questions to ask, so you should be able to answer, 6 or 7, and then explore more. And at least you see that whether your idea is going forward in form of product or in form of services which can be sellable in the market. Also, if you get the idea, you should also do these kind of checklist analysis first, that okay, your idea is good, but then is there a need for that idea? If you are, let's say, developing something which people don't even need, then it doesn't make sense. Also, even if need is there, what is the cost? So for example, if you are bringing, let's say, a shaving razor, and if there is already a Gillette razor available for 20 rupees, and you are launching something for, let's say, 500 rupees, so who's going to buy it? So that is also very important. Also the target customer. Let's say if you're trying to sell a Mercedes or Audi in a small town, for example, tier 3, tier 4 cities, perhaps you won't find so many customers. But if you do that in Delhi, then of course that is the case. And that is why you may have seen that these automobile companies, they do have their showrooms very, very strategically placed. Similarly, you take an example of Starbucks coffee. They sell a coffee for almost 300 rupees. If they do it in Delhi at Connaught Place, you can see that people are flocking, is still waiting, and then always there are 20-30 people waiting because there are customers. But if you open a Starbucks cafe in a village, nobody is going to buy a coffee worth of 300 rupees. And also the segment of market itself, like for example, there's a Honda car, there's a CRV, but there is also Amaze, there is also Brio. So that also need to figure out. And then what are the activities you require to produce your product? What is your go to market plan? And also how your business will become profit making. This is also very important. It can be interesting, it can be a very exciting idea, but if it is not making any profit at the end of the day, it will not be sustainable. It will be just, it will become an academic exercise. So you also have to make sure that it is sustainable and it brings some revenue. um, there is a new concept, not so new now, but for India, there are many incubators, and they got established. Most of them got established in last, I would say, 15 years or so. And there are multiple opportunities. If you have an idea, you go and talk to an incubator, and they will tell you that what are the policies, that whether you can incubate your idea or not. And even before you move to incubator, you should have a pre-incubation, like development of prototype, writing business plan, do the analysis, what are the strengths and weaknesses and opportunities and threat to your idea, like a typical SWOT analysis. You can find it online or in typical management books. We are also an incubator at IISER Bhopal, although I assume that most of the students who are attending this talk are from Delhi. But yeah, if you want to discuss your ideas with us, or you do consider a possibility of virtual incubation, we are happy to talk to you. We are in the main building, administrative building at IISER Bhopal at second floor, and you can write to us at iice@iserb.ac.in. Generally, an incubator provide you the facility, a space to set up your lab, to set up your office, and also they have the common facility, for example, state of the instrumentation, and they sometimes they provide financial assistance in form of seed funding, also the networking with investors, how to raise money, how much money do you need, how to make your company overall successful. Overall mentorship every incubator provide. I'm sure there are incubators at IITs and at regional universities, and we also have an incubator which is funded by Department of Science and Technology. They very generously funded us. Also, this is another thing which I like to mention, that a lot of time people want to start companies in bigger cities. For example, Bangalore, Pune, Hyderabad, Delhi, NCR. Most of the startup, they are restricted into these areas, but generally what happen that operational costs in these areas are very, very high, and that is why now tier 2 cities are emerging as the major hotspots in the country for the startup growth, because your cost of operation, your cost of failure, even hiring manpower, everything is much cheaper. And there was an analysis done that the same biotech company, if you run in Bangalore, in the same money, you can run the company in Bhopal or some other place for, 2 times more duration. So like you will be finishing that money or capital within a year in Bangalore or in Pune, but in tier 2 cities, you can run the company for 2 years, 2.5 years. But it depends. Every place has its own positives and negatives. But yeah, tier 2 cities are emerging as a major hotspot in the country for the growth. Are you eligible to start a company? Very likely, yes. And I would say if you have an idea, please do walk into the incubator or write to them. Or if you want to write to us, most welcome. We do have a policy in place where you can start your company. And wherever you go, wherever you start, you should always ask for the entrepreneurship policy. You should always ask, if you start a company, how much money that you need, how much equity or share you will be diluting to the incubator, or if there are investors coming, then how much equity you are giving to them, or revenue sharing. These are some of the questions that you should ask. We have recently finalized our entrepreneurship policy, which has been recently approved by Board of Governors, and it is pretty much in line with different IITs and national institutions across. This is a typical incubation methodology. You submit your proposal, then they screen it, then there are evaluations and recommendations. Then they tell you that, okay, that is the money they will charge from you. And then they finally sign an agreement, and then you are incubated. And then you are incubated for a duration of, let's say, 2 years. And then after 2 years, when you grow and you are at the mature stage, then you can take the exit and can move to a separate place now. So that is another that is the typical methodology incubators use. These are some of the startups which are already incubated at ICE. This slide just to give you the perspective that these are the companies which were started mostly by faculty members, also generously supported by government through different grant schemes. And also there are a couple of startups coming up now from students, and they will be starting some of the company. 2 or 3 students have already started with us, but now the more traction is coming up, and hopefully we will have another 6 to 7 startups. So very likely maybe 15 by end of next year. These are some of the events that we conduct, and you are most welcome to join us through our YouTube channels or through emails. We do invite very successful and young entrepreneurs from across the globe. and also keep doing events on IPR, on innovation, on technology transfers with different incubators, with different entrepreneurs, business leaders, people from different companies, and also do have competitions where you all can participate. Like Curveball is coming up. I think the deadline is September 24th, so you can also register as a student if you have some other idea, and we can provide the complimentary incubation if you are successful. So I would like to end my presentation with this slide. If you're thinking that, oh, you are too young to start, you are still in a college, you still have time to graduate, fine, that is a good idea. But then maybe you will be doing a job for 4 or 5 years, going abroad to study, and then maybe you will think of starting a company. Perhaps then that is not maybe the right approach, or that might be the right approach. But I think if you are too young, it should not restrict you to start a company right away. And I always quote this example of Kylie Jenner, who started this company, Kylie Cosmetics, and her real time net worth is almost more than $1 billion. This is the data of 2019. And I'm sure it is even, it get better now. So at the age of like 23, 24, she's already a billionaire. And I also would like to mention that entrepreneurs do contribute for the societal development. And in fact, they contribute at a very, very larger scale. If you look at these foundations like Bill and Melinda Gates Foundation, Wellcome Trust, Azim Premji Foundation, Ford Foundation, these are the foundations. They donate for science, they donate for research. And how do they donate? Because they made so much of money that part of the money they now they kept aside in form of like I would say a trust which provide funds for research. And not only that, but even the Nobel Prize, which is also endowed by the money given by Alfred Nobel. Alfred Nobel was also although he was trained in science, but he was also an industrialist, and that's how he made money, and he made the endowment for the Nobel Prize. With that, I would like to end my presentation, and I would be happy to take question, and would like to leave this with the encouragement that entrepreneurship is a very, very exciting area, especially for the modern India or modern times in India, and you should seriously think about it if you have an idea. And thank you very much for having me, and I'll be happy to take.
Thank you, sir. So we have one question in the chat box. Most of us belong to middle class families. Starting a business or corporation appears to be out of our reach. How to overcome these assumptions and fear of failing when there are much bigger, larger corporations with higher resources?
Well, I think if your idea is good, and if you see that most of the corporations who are bigger, some at some point of time they were also from a middle class. They were in fact were from a lower middle class. It's just that sometimes it takes one generation, sometimes it takes two generations. I'll give you examples of some of the very large companies in India. Reliance, which was started by Dhirubhai Ambani. So he was not a very rich person. He was very poor. Okay, next generation is richer. That is fine. And they are the richest now. Similarly, if you think about Wipro, Azim Premji, his father was rich, but not as rich as he is. And the way he built Wipro into software. Earlier, Wipro used to make a lot of cosmetics product and soap and all these things and oil and all, right? But now it's a software company. Infosys was started by people who went to IITs. They graduated from IIT and they were not rich. They were from a very, very, I would not even say middle class, like I would say lower middle class people. And in fact, Narayan Murthy was supported by, 10,000, 15,000 rupees. He borrowed it from his wife. And that's how they started Infosys. There's so many examples. Biocon, Kiran Majumdar Shah was not from a very rich background. She started the company in garage, and now you see where Biocon is. Even now, Biocon has started multiple companies. If you see Syngene is there, they acquired even more companies. So there are so many examples like that. I don't think that they came from a very rich background. And second point I like to add here, that earlier it was even difficult to start a company. Now there is a framework and network of investors available. So if your idea is good, if you think that it is genuine, it can make some impact in the market, then there are people who are ready to fund you. Of course, they will take some shares in the company. If they give you, let's say, 50 lakhs, they may ask that, okay, give me 10% of the company or 20% of the company, but you get the money of it. So there are so many possibilities. Your idea should be good. I don't think that if you are from middle class, that will restrict you. In fact, that makes you better. That can make you better entrepreneur, because you will value money. You know that you have to make money, and you are deprived of it. So you will want it more, and you would like to make it success story, right?
Okay, so we have another question in the chat box, but I believe that you're short of time. So,
No, I'm fine. I can
all
still
right.
take it. My meeting starts at 12, so that is fine.
All right. So another question is Naveen Garg asks that he wishes to learn digital marketing and apply it with his science background. According to you, how can he do that, and what are the opportunities of digital marketing in science application?
So are you looking for a job? Then I will have a different answer. But digital marketing, like I did not quite get the question, but if he could clarify, or else I'll try to answer. So like in digital space, a lot of things are happening. So there are multiple startups came up who are just using the information and then spreading it for the marketing purposes. There are multiple startups coming in the PR, like public relations as well, and in also in line with science advertisement, there are startups and companies. One or two I know, they are doing science PR. So, for example, let's say they have very specific people who can take laboratory science, write it in a layman language, and publish it for the general people. So that is one of the space which has lots and lots of opportunities. Similarly, there are edutech companies which are coming in the digital space and digital marketing space. So you can also tap those as a potential client and customers. And if you can clarify a little better what exactly you want to do, perhaps I'll be able to answer it better. You can write it to us.
We
Okay.
will request actually Naveen to mail Dr. Hussain, and he will be happy to answer your questions.
Yeah, please.
As already mentioned. So here I would like to give vote of thanks. Thank you, Dr. Hussain, for your wonderful lecture. It is indeed intriguing for us to know that how even scientists can not only impact science in life, but also economy in a big, big way. So thank you, sir, for the knowledge-packed lecture. And I'm sure that the takeaway from this lecture would be to translate the science we do and try and impact masses. Thanks again on behalf of everyone for sparing your precious time and enlightening us with your talk. I'm sure that all of us have learned from your lecture your patience and virtues.
Thank
I
you
would
so
now
much. Thank you so much. I hope I finished on time, and thank you for the opportunity. Thank you.
Thank you for such a wonderful presentation, sir. I would now request Dr. Roshan to now take over. Roshan.
Thank you, Dr. Jaswinder. Our next speaker is Dr. Utkarsh Sood. Dr. Sood is working as a research associate at the Energy and Resource Institute, New Delhi. He is the director and co-founder of Fiction Private Limited. He completed his graduation, post-graduation, and PhD from University of Delhi. During his doctoral research, he worked on the complete and comparative genomic analysis of clinical and non-clinical isolates of Pseudomonas aeruginosa. He was awarded the PhD degree by University of Delhi in November 2018. He is a young researcher with 25 publications, including peer-reviewed research, review articles, and book chapters. He has recently contributed 2 genus chapters in prestigious Burgess Manual of Systematics of Archaea and Bacteria. He is now enthusiastically working in the field of comparative genomics, microbiome studies, and secondary metabolites of bacteria. Over to you, Dr. Utkarsh.
Okay, good morning, everyone. Thank you, Dr. Roshan, for the wonderful introduction. And I would like to thank the organizers for giving me this opportunity to present a very short presentation that I have made for the young participants. That is how microbiology is playing a very good role in achieving sustainable development goals. Okay, so I'm just sharing my presentation. So my screen is visible. Hello.
Yes, it's visible.
Yes, it's visible. Yes.
Okay, thank you. So, all right. So, to all the young participants, this is a very short and crisp presentation that I've made to elucidate the role of microbes in sustainable developmental goals. Okay, so as I am Utkarsh, I'm working as a research associate at the Energy and Resources Institute, New Delhi. So, in school, we all have learned about sustainable development. So, basically, the idea behind sustainable development is that the development that meets the need of the present without compromising the ability of future generation to meet their own needs. So basically, we all know that we have resources on Earth, like fossil fuels, we have minerals, and we are utilizing these resources for our development. But nowadays, what the current generation was doing, that we were over exploiting the current resources, and that would have led to pollution, number one, and number two, it will compromise the ability of the future generation to meet their needs. So the prescribed scenario or the best scenario is that we have the resources in such a way that the waste is recycled and we have greenery on Earth with low poverty, renewable uses of sources of energy, as well as prosperity on Earth. So this is the current scenario that people want to achieve by using sustainable development. So to achieve this, United Nations, they have proposed 17 goals that can help us to achieve sustainable development, and therefore they are known as sustainable development goals. So the first goal is no poverty. So we should reduce poverty on Earth, zero hunger, we should have good health and well-being, the quality of education should increase, there should be gender equality, we should have clean water, affordable clean energy, decent work and economic growth, industry and innovation, reduce infrastructure, reduce inequalities, sustainable cities, like that, and life on land, good life on land, peace and partnership in institution to achieve these goals. So basically, what now scientists have learned, that microbiology has an integral part to play in achieving all these 17 goals. So now moving forward, so as you can see that the United Nations has put forward these 17 global goals with 169 targets. So we are just dealing with these 17 goals. They have 169 targets, and these 17 goals were adopted by all the members of the United Nations in September 2015. So these to understand them in better scenario, how they will benefit the people, how they will benefit the planet, and how they will promote peace on Earth. Basically, we can see that 29% of these 17 goals, that is 5 out of these 17 goals, are basically directly linked to the people of the planet. 29%, that is again 5 of these goals are linked to the prosperity. One goal is linked to the peace on Earth. Second is one is for the partnership between different institutions to promote science, to promote well-being, and to promote development. And another 5 goals are basically for the planet. So we have 5 goals in planet, people, prosperity, peace, and partnership. But to understand them in a better way, we can club these goals into 3 basic pillars. So the first pillar is economic pillar, where the economic scenario of Earth is taken into care. The second pillar is environmental pillar, where the environment is taken into care. And the third one is the social responsibility pillar. So now you can see I have divided, or the United Nations have divided these 17 goals into 3 main pillars. And we will be taking 3 examples to understand. So for each goal, you can see and you can study that microbes are playing a persistent role for achievement of these goals, and they can play further beneficial roles in that goals. But I will just touch upon 3 major goals, and by incorporating 3 scenarios. So the first is economic pillar. So to understand how economic pillar of Earth is maintained by microbes, we will take an example of antibiotic resistance and good hygiene practices. Okay, and in the case of environmental pillar, we will touch upon the clean energy and organic waste management. And for the social pillar, we will re-emphasize on the need of microbiology literacy in society. So the first example, that is the example of antibiotic resistance. So as you can see that we all must have heard about antibiotic resistance, how it happens. So basically, this is a very simple diagram which depicts that how this phenomenon spreads in microbes.
So as you can see, as we know that each organism has a genetic repertoire, and some bacteria might be drug resistant. That means they are not susceptible to the particular antibiotic. So in the case of an infection, there may be a colony of bacteria which is a resistant bacterium. Okay, and when we treat this colony with antibiotic, what we will see that this one bacterium remains. Okay, and this can then multiply to form more colonies. And now we have a problem of drug resistant bacteria. The drug that was working previously on majority of the infection caused by a particular bacteria will not work here in this scenario when the entire community of bacteria has become drug resistant. And this drug resistance can be transferred onto another bacteria by this bacteria by a phenomenon known as horizontal gene transfer. So now moving forward, as you can see, what is the mechanism between the drug resistance? So as you can see, that drug resistance is basically because of the genetic repertoire of bacteria. So we call this genetic repository or the genetic repertoire as resistome. That is the collection of all genes that directly or indirectly contribute to antibiotic resistance. So basically what is happening, some genes in the genome of the bacteria are getting inactivated. So if the gene becomes inactivated, it can help the bacteria to become resistant to antibiotic. Okay, so we need to understand the mechanism also in brief. So I'm not going into details because I know that very young participants are participating in this workshop or the series. So basically inactivation of genes, that means now the antibiotic will not work. And sometimes what happens, certain genes becomes hyperactivated. Okay, or sometimes the target of the antibiotic is not there in the bacteria. So in that case, the bacteria will become antimicrobial resistant, and we will have colonies of this bacteria having more and more infections. So the problem is that this can be transferred, as I've already told you, from one colony of bacteria to other colonies of bacteria by mobile genetic elements as well. So the mechanisms include production of certain enzymes that degrade antibiotic. Okay, so these enzymes can degrade the antibiotic, and that antibiotic will not have any effect on the bacteria then, or they will alter their sites where the antibiotic binds in the structure of the bacteria. They can alter the site, so that is known as target site alteration, or they can produce more antibiotic targets. So there are always targets on the bacterial cells to which the antibiotic will bind, and if the bacteria is producing more targets, then the concentration of antibiotic needed to kill that bacteria will be needed in high concentration. So that cannot be given to the patient. So in this way, the bacteria escapes, okay, from the antibiotic pressure. The last mechanism is that they can throw away the antibiotic from out of the cell. Okay, so they can throw out the antibiotic, that is known as efflux, and then the antibiotic will not have any effect on the bacterial cell. Another important aspect of understanding how microbiology is playing important role in our health, our prosperity, is that there are certain infections that has the ability to jump from one host to another. So you must have seen, and we all know that COVID infection or the COVID disease that is caused by SARS coronavirus 2 is basically a virus that has jumped host, and now it is causing havoc in the human population. So similarly, there are certain bacterial strains as well that can jump off from the organism, one organism like humans, to another organism like pig. There they can acquire resistance and then reinfect the people and become superbug. So what is a superbug? Superbug is basically an organism that is resistant to majority of the antibiotics that are used in clinical practices. So not only in humans. So basically, if you are thinking that this problem is only in humans, that is not the case. We all know that we eat poultry products, or we eat meat products. So these organisms, these animals are also given antibiotics in high dose so that they don't catch diseases. So even if the high dose of antibiotics given to these organisms, then also the resistance can be acquired in the animal host and then passed on to the human host. So this is just an overview that almost 30 million pounds of antibiotics is used worldwide in pig population. Okay, so to rear or to basically in pig farming, they are using 30 million pounds of antibiotics, which is a very high number, and that can lead to the promotion of antibiotic resistant microbes in pigs. Okay, and when the human consumes such meat, this problem can be passed upon to one host to the another. So now moving forward, so this is just an example slide that certain bacteria residing in certain hosts or certain habitats. So hospitals, as we know, is a source of superbugs like Pseudomonas aeruginosa and Acinetobacter baumannii. So these two types of bacteria are known to be extremely drug resistant. That means they have gained resistance against almost the entire set of antibiotics that is known to humans. Okay, so we are going back to the pre-antibiotic age. That means there will be no antibiotic to treat the infection caused by these two bacteria if they are present as a extremely drug resistant strains of these two bacteria. These bacteria can jump from the hospitals like Staphylococcus pneumonia. These infections are very common. When a patient gets admitted to the hospital, he or she can catch that infection and then spread it in the community. Similarly, from the poultry, from the cattle, from the livestock that people maintain or eat, certain bacteria, as you can see, the genus of the bacteria, they can switch hosts and cause diseases in our system as well. So this slide basically depicts how antibiotic resistance is hampering the sustainable achievement of 5 sustainable development goals. So as you can see, if we have to generate newer drugs or we have to give high amount of drugs, then the person need to buy those drugs, okay, and he or she cannot work. So this AMR or antimicrobial resistance could push an additional 24 million people into extreme poverty by 2030. So in coming 8 years, because of AMR, poverty will increase, okay. And as you can see, the livestock production is decreasing because the problem of antibiotic resistance is increasing in livestock. So the poultry or the pig farming is getting hampered, and the production and the yield of meat that is being generated yearly is getting reduced. So 7% loss is almost every year is there. So we cannot maintain good health and well-being, okay, if the problem is there because of the antibiotic resistance, and leading to reduced inequality. So where the problem is persistent, that country will not have proper development, and that will lag behind the other countries where proper development is going on, decent economic growth is going on. So to keep this antibiotic resistance in check, we should always keep in mind that we should be knowing microbes, which microbe has the ability to cause which diseases and where they are situated, how antibiotics are to be used rather than to be misused. Okay, so this was the first example that I have taken up in the case of antibiotic resistance. The second example is basically regarding your clean energy and solid waste management. So as you know that lots and lots of organic waste is generated on earth every day. We have livestock, we have crops, we have wastewater, we have food waste. So all these waste is generated, but the good thing about this waste is that it is biodegradable. So if we properly degrade this waste and instead of degrading it, convert this waste into an asset or a form of energy that can also help us to attain sustainable development in near future and will help us in achieving these 6 sustainable development goals. How? One thing, using the help of microbes, we can digest all these waste using an anaerobic digester. And from this anaerobic digester, we get biogas, we get digestate, that is the remain after the digestion, which can be converted into fertilizers, soil amendments, and livestock beddings. Okay, so this digestate is also used. We can convert biogas into heat or electricity, and we can also convert this biogas and purify it into biomethane to run cars as fuel and also light up the gas grid. So as you can see, the waste can be converted into asset or can be converted into renewable form of energy by using the help of microbes, which can help us in achieving 6 sustainable development goals. So now moving forward, the next social pillar. So how social pillar is important and how microbiology literacy can play a very important role. So the common perspective that is being taught in ads as well, that microbes are bad for humans. We always, when microbe word is used, the first picture that comes up in our mind is germs. Okay, so basically microbes are not always bad for the human. There was a study in Italy in which 60% of the children, they said that microbes are harmful, and only 25% admit their positive role. So this perspective we need to change. Microbes are our friends also. Okay, so the gap of reality, that how microbes are and how they are, how and what other things, good and beneficial things they are doing for us, need to be taught in the school. So as Rupla sir was explaining in the lecture, that this is the time when we need to incorporate A for Acinetobacter, B for Bacillus in our school curricula, and make students understand that how to use microbe for our benefits. So this will help us to have quality education, have gender equality, have peace, and have institutions who can correspond and transmit the information that they have gathered among themselves. So the take-home message for all the younger generation that is listening this lecture is that we should not take antibiotics without prescription. Okay, in India, this is a common, we can say, outlook, that we just go to a chemist, buy antibiotics, and take it according to our perspective. Okay, without consulting a doctor. So antibiotics should always be taken up after a prescription, after prescription by a registered doctor. Okay, second thing is that microbes are beneficial for us as well. As you have seen, they are a source of antibiotics, they are a source of biomethane, fertilizers, and they also degrade the solid waste. If microbes are not there, you will find all the organic waste lying here and there on the earth. Third, not all microbes are bad, but we must follow proper hygiene to restrict the growth of disease-causing pathogens. Okay, so you should know this, that we have to have a continuous battle, but only with the microbes that are pathogens, and we can maintain them and we can restrict them by only having proper hygienic environments. The last take-home message is that microbial literacy, it should be included in the school curricula, and once a person is known about the microbiology and how it affects the overall development of the society, that can change our developmental goals. So in the end, thank you for your attention, and this is a very important slide where it has been shown that microbiology can link all the 17 sustainable development goals, and without microbiology you cannot achieve sustainable development, that is for sure. Thank you very much.
Thank you, Dr. Utkarsh, for this wonderful talk describing microbe to think as an alternative to meet the global goals. I do have a question. What is your opinion about this? If we want to inhibit certain pathogen with the help of good microbes, so can it meet any sustainable development goals?
Yes, sir, because it can meet, I think it can meet more than one sustainable development goal. Number one is for good health. Number two is for economic growth. That is, if we can maintain or restrict the pathogen, we don't need to take medicines. Okay, so like in the previous lectures, we have heard that if you maintain your lifestyle, if you train your microbes, if you take probiotics, if you take good food, if you do exercises, then the good microbes will increase in number in your gut, and they will automatically do their job and restrict the pathogens.
Well said, Dr. Utkarsh. Thank you once again for this lovely lecture. Now I hand over this session to Dr. Nitish. Over to you, Dr. Nitish.
Yeah, thank you, Dr. Rosen. Now proceeding with the program, next talk entitled Microbiology as a Career Prospect will be delivered by Dr. Gauri Garg Dhingra and Dr. Rosen Kumar. Today, I will take this opportunity to introduce both the speakers. Dr. Gauri Garg Dhingra is currently working as Associate Professor at Department of Zoology, Kirodimal College, University of Delhi, and has more than 15 years of teaching experience at undergraduate level. She has completed her graduation from Sri Venkateswara College, then did B.Ed from CIE, University of Delhi. She has done MSc and PhD from Department of Zoology, University of Delhi. During her doctoral work, she has extensively worked on genetic manipulations of rifamycin gene clusters in Amycotopsis mediterranae. She has also worked as postdoctoral fellow at Department of Zoology, University of Delhi. She has many publications in peer-reviewed journal in her credit, and also has organized various workshops, conferences, and courses at national and international level. I welcome you, ma'am. Today, also with us, Dr. Roshan Kumar, who is currently working as assistant professor at the postgraduate department of zoology, Magad University, Bodh Gaya, Bihar. He has published more than 30 research articles in peer-reviewed journals with more than 500 citations and IH index of 15. He has completed his PhD under the supervision of Professor Ruplal at the Department of Zoology, University of Delhi. During his PhD, he was a visiting researcher at Helmholtz Center for Environmental Research, Leipzig, Germany. After his PhD, he was a Land-Law-Led postdoctoral fellow at the Department of Veterinary and Biomedical in South Dakota State University, USA. His work mainly focuses on genomics and metagenomics, uncovering the environmental and gut ecological aspects of microbiology. Thank you, Dr. Gauri and Dr. Roshan, for taking out your time from your busy schedule and agreeing to share your thoughts with us. I invite both of you for delivering your talk. Over to you, Dr. Gauri and Dr. Roshan.
Thank you.
Thank you so much, Dr. Nitish. That was a wonderful introduction for both of us. Now, good afternoon to all the participants. Today, we are here to present the International Microorganism Day on 17 September under the aegis of INSCAR International Outreach Program, MICROSPHERE.
Our aim is to spread awareness about the microbes which are present everywhere around us and affect almost all aspects of our life. Our mission is to advance the understanding and the impact of microbiology by connecting and empowering communities worldwide.
I am Gauri Dhingra from Crodimal College, Delhi University.
I am Roshan Kumar from Magad University, Bodh Gaya. We together will try to make you understand how to become a microbiologist if you choose to be the one.
So, we have all learned the ABC of languages, mathematics, social sciences, and basic sciences since our primary classes. And now we need to know what is in store for the future.
A day comes when we need to decide आगे क्या करना है.
And our options are science with maths, science without maths, commerce, arts, or फिर job किसमें?
साइंटिस्ट बने तो क्या हाल होगा?
क्या ये हाल होगा? Okay, let's find out what are our choices. Medical and non-medical या फिर कुछ और भी. Is biology an isolated science? Roshan, please let us know.
If biology comes with geography, it is called biogeography. If it comes with physics, it is known as biophysics. If it comes with biology, if it comes with mathematics, it is known as data analysis. If it comes with chemistry, it is biochemistry. And if it comes with economics, it is bioeconomics.
Oh, that's interesting. So where can we lead with these choices?
We can go to medical sciences, allied medical sciences, biological sciences, and pharmaceutical sciences.
And what jobs can we get?
I can go to industry, improving crop health, involved in scientific writing, become an academician, a professor, as our speaker today, or any high school teacher, work in government for making policies or law, you can say, work for conservation biology, some non-profit organizations such as museums, botanical gardens, etc.
Oh, that is nice. But if I choose to see and study microorganisms, then what am I supposed to do, Roshan?
Well, I would need a degree in microbiology or any biological science and can go to industry, research, and clinical biology.
So you mean to say microbiology can be the basis of biotechnology, molecular biology, clinical pathology, medical microbiology, and many other applied sciences. Confused? Let's understand.
Yes. What does a microbiologist do? The question is very common. During the course, the microbiologist or biologist studies about microorganisms, bacteria, fungi, algae, viruses, and pathogens. They study how microbes live, grow, and interact with their environment and affect human health.
So, to become a microbiologist, I think we need a background of biology, a degree of microbiology, or for any biological science.
So, what is the path to become a microbiologist? As you can see in the slide, if you are passing your 12th standard with biology, you can choose for BSc Honors Microbiology. Then later on, you can opt for MSc Microbiology, and then you can have the degree, PhD degree. If you choose to be in any other biological science, then you can opt for microbiology in your master's, and then you can go for PhD. Or you can go for any other diplomas, or directly from a biology background, you can directly go to MBBS.
Okay, then what can I become? I can become a clinical laboratory scientist, a clinical microbiologist, a microbiological analyst, microbiological laboratory technician, a microbiologist, microbiology director, microbiology laboratory manager, quality control microbiologist, study director, bacteriologist.
You can also become a microbiologist. After your PhD, you can learn molecular biology. You can be a clinical microbiologist. You can be an agricultural microbiologist. You can also enter in medical microbiology. You can be a researcher and you can discover a vaccine. You can be a pharma researcher and so many things.
Oh, that's wonderful. Okay, that means anyone can become a microbiologist, whether they have been physicians, doctors, or chemists, like Alexander Fleming, Louis Pasteur, and many other famous microbiologists.
Here are some of the institutions in our country which offer degree courses in microbiology. These are Gargi College, University of Delhi, Shahid Rajguru College of Applied Sciences for Women, the Oxford College of Science, Presidency College Chennai, Perul University Vadodara, Ferguson College Pune, St. Xavier's, and so many others. These are the options that you can opt for BSc courses in microbiology.
You're forgetting your own university gives that course, Magadh University. Yes. And then where do I get the master's degree in microbiology? Oh, these are IITs, JNU, DU South Campus, Jamia Millia Islamia, Madras Christian College, Manipal University, Amity University, Sri Venkateshwara University, Noida International University, Swami Vivekananda University, so many. And what about PhD?
So PhD is offered by several other institutions such as Annamalai University, University of Delhi, Tamil Nadu MPhil Microbiology Center, GTAM University, Vishakhapatnam, IITs, and all ISER institutes.
Oh, that's a long list. But then I would like to know, what are the various entrance courses for microbiology, Roshan?
For graduation, you can opt to Osmania University. They offer microbiology entrance test. They call it OUCET. Then we have Goa University for microbiology. We have Mahatma Gandhi University common admission test. Then for post-graduation, we have Indian Institute of Sciences, Bangalore, then Indian Institute of Technology, then Goa University. So all these places offer entrance for microbiology for the course of graduation and post-graduation.
So we have many common tests for which a student can sit for microbiology exam. And then, Roshan, what are the roles and responsibilities of a microbiologist?
So once you become a microbiologist, you need to do something for the society as a mandate for your role and responsibilities. You need to provide laboratory services for health departments for diagnosis and treatment. You need to investigate the relationship between pathogen if something comes, such as epidemic or pandemic. You can use bacteria and microbe to develop vitamins, antibiotics, and several other compounds that can meet the sustainable development goals. You can also prepare technical reports and recommendation based on what you study, what your project gives you. And overall, you can use a variety of specialized equipments to infer the conclusion from your studies.
Oh, that's wonderful. So it is at the end you finally decide who you are. So we wish all the students all the best for their future. With this brief introduction to how you can become microbiologist, we will sign off for now. And now I think don't be afraid of moving forward. The future is very bright. Am I true, Roshan?
Yes. Thank you. Thank you very much.
And now we move on to the next session. Over to you, Nitish.
Thank you, Dr. Gauri and Dr. Roshan, for this informative and interesting talk, in which our young participants get an exposure of various career opportunities related to biology and microbiology, which they can pursue after higher secondary level. All school and the college students must have benefited from this informative lecture. Now I invite Dr. Charu Tripathi, who is working as an assistant professor, CMP Degree College, Allahabad University, to coordinate the question and answer session. Over to you, Dr. Charu.
Thank you, Dr. Nitish. We would now like to take up the questions by participants. Deji Kumari's doubt is that she belongs to a middle-class family and wants to become a doctor. Same doubt is raised by Varsha Rani, who wants to become a cardiac surgeon. So they want to ask Dr. Roshan and Dr. Gauri about what path they should take. What should they do after school? They are currently in school.
Okay. Dr. Gauri, you may like to answer this.
Roshan, please do the favors.
Okay. So if you are in 12th standard, you need to opt for biology. And then you have to prepare for NEET examination. And once you score, then you get some college. And for doing so, if you are from a middle class and if you don't have any background, if you qualify for NEET examinations, NEET exam, then there are a loan facility available. Banks can offer you loan in order to complete your study. Later on, you can meet all those expenses when you become a doctor.
And I would add, you can do MD in microbiology as well.
Okay, thank you, Dr. Gauri. Andalip Anjum also has a doubt. He wants to know the scope of microbiology and the job opportunities which can be taken up after studying microbiology.
I think we have listed that in the presentation. There are, first of all, inspired by the talk from Dr. I think Amjad, you can become an entrepreneur. Okay, even if you don't have the resources for it, right, Roshan? And then there is a list of opportunities that we have just shown. You can become a scientist, you can become a school teacher, you can become a college professor, you can become a researcher, and there are so many other options. You can go to industry, you can also work for non-profit government organizations, you can also be a part of government policies and laws, and many more.
You can also enter in specialized fields such as clinical microbiology, agricultural microbiology, and you can work on medical or non-medical fields depending upon your expertise. So at this point, what you need to do, you need to specialize yourself based upon your interest and skills.
And the best part is not just you can work for microbiology. If you are a microbiologist, you can work for any other allied sciences. Any other query, Charu?
Yeah, Swati Rai, who's a BSc student, has an interest in entrepreneurship and would like to balance in both studies as well as in business. So she wants to know how science can benefit her in pursuing her entrepreneurship goals.
So if you are interested in your studies, so what you can do, you can frame a question. And based on that, you can get certain unique answers. I'm talking about certain biological hypotheses. Based on that, you can infer certain results. And using that as an idea, you can enter in the field of entrepreneurship. So if let us suppose you have a microbe that is doing something amazing, and it is producing certain compounds, which is very unique and costly. What you can do, you can use that compound to write certain grants. As for example, there are certain incubation centers, as you have seen before. They are providing monetary help in order to boost your idea. And finally, if you come up with certain product, what you can do, you can start you can exit from that incubation and then you can start your own company or a startup.
Anshu Rana wants to ask about the future prospects of biomedical sciences. The student is currently in BSc and wants to pursue masters in biomedical sciences. So can you suggest some institutes or universities which offer this course?
So we have ACBR, that is a part of University of Delhi, where this biomedical sciences is being offered. You can also search in University of Hyderabad. And most probably there are so many options. We cannot tell you right now, but yes, there are institutions which offer these biomedical science courses, and depending upon your field which you will enter after your graduation, that will decide the very purpose of which field you want to opt for your biomedical research.
Yes, that's right.
Nishan Thakur's question is that what are the prerequisites for doing MD in microbiology?
You need to have an MBBS degree to enter into MD.
Kalpana Jha asks that what is the procedure of getting into PhD and how long does it take approximately?
Well, 3 years of graduation, 2 years of masters, and I would say 4 to 5 years of PhD. So it rounds out about 10 years.
So the best part is you need not to be depending upon your family for getting your PhD. What you can do, you can clear entrance examinations that can provide you fellowship after your master's degree, and you can do your PhD, because nowadays there are JRF, SRF, which gives you handsome salaries. So what you can do, you can opt for PhD if you get all these fellowships.
And there are certain research projects running in various laboratories where you can apply as a junior research fellow as well, if there is a provision.
Kalpana Jha would like to ask you if you recommend MSc PhD integrated course.
Yes.
Yeah.
So there are certain institutions which generally provide this type of courses. So what you generally prefer, why you prefer generally this integrated course? Because from masters onwards, there is a semester where you basically have to submit a certain, what do you call, dissertation. So that dissertation can be the foundation of your PhD. And while doing so, it will save some time for you because MSc PhD on an average, it will take 6 years. But if you will do it on integrated basis, it may be over in 5 years also. So that can be one of the advantages.
One more advantage is if you're doing your MSc PhD integrated course, in your final year of MSc, you will be given exposure to a laboratory and you will be learning many microbiological techniques which will be helpful to you during the PhD process. So definitely, you should go in for an MSc PhD integrated course.
Ananya Mishra has a doubt whether microbiologists have opportunities in the R&D sector.
Yes, they have a huge possibility in R&D sector. As Roshan said, they can go in for many kinds of genetic engineering projects. And all these genetic engineering and molecular biology and biotechnological projects are ultimately related to the R&D industry.
Ankit Kumar asked about internship programs in microbiology and for MSc students.
Yes, there are certain programs which gives you internship fellowship, as well as they basically provide you the centers where you can go and have that internship. And during that, they pay you some money as well. So there are things. I think CSIR is also providing such training fellowships.
Even DRDO, you can look for. There are many centers you can search online.
Okay, Himanshu Jain asks about the procedure of becoming a clinical microbiologist.
You have to work on clinical microbes. So that will start from your master's, because if you have interest in clinical microbiology, what you can do, you can choose your dissertation in master's dissertation in clinical pathogens, and then you can opt your PhD. and you are a clinical microbiologist.
And you can also go via MBBS. You do your, you do your doctorate, and then you can go in for clinical microbiology. That is also an option. Pathology, in fact.
Vanya Garg is currently pursuing masters in microbiologist. She wants to know that can you suggest some labs where she can do her dissertation, preferably the labs which would let her do the research for free?
So most of the labs, they generally won't charge any money, but you need to personally communicate with the supervisors from all those central universities or institutions. You can drop a mail requesting them to guide you through your dissertation. So they will give you the space as well as the resources to complete your dissertation or training, whatever you want to do. That is free of cost in most of the institutions.
And many of the supervisors are looking forward for such students who are interested to work for them. Additionally, you can get publications as well.
I think Ankush Singh's query is also answered in the same question. He wants to know that what is the way for joining the labs for research during MSc and PhD?
Yes, you can directly communicate with the supervisors, and then they will let you in but there should be a proper way of writing the mail. You should attach your CV with that, so that they will come to know about you first and then your interest, and then they will give you the permission to work in their laboratory.
In fact, many of the institutes, they advertise their internship programs as well. That can be also found.
Yes.
Okay, and Swati Rao has a very relevant question that because of COVID, they could not gain much of the practical knowledge that they would have gained if they had been in a lab or they would have been attending regular college. So what can you suggest? How can they enhance their practical knowledge in this scenario?
So you can become a bioinformatician. So bioinformatics does not need much of all these wet lab experiments, but at the same time, you can gain insight into any living system, knowing about different proteins, their function, different genes, different enzymes. So bioinformatics can provide you extra edge, keeping in mind the current COVID scenario. There are certain courses which is being provided by Fiction Private Limited. You can be part of one of those courses, because that particular company is one of the partner in this particular event. So they are basically bioinformatics based organization which helps you to learn so many things, that helps you to understand the basic system.
And when this sad situation is over, you can always work in a laboratory with one of the supervisor and improvise on your techniques and the practical knowledge.
Thank you, Dr. Gauri and Dr. Roshan, for taking up so many questions, for highlighting the career prospects of microbiology, especially for our young generation present here. I would now like to invite Dr. Renu Solanki and Dr. Shelly Anand to introduce the next session.
Hello, that was a wonderful session, and I think students were waiting for that session to happen. And the questions are still flowing in. Students, due to paucity of time, we'll have to take forward the next session. And this is a hands-on session where we have with us the students of life sciences, undergraduate students. We have Ankita and Yogita, who will be taking up this session. They'll be guiding you how easy it is to isolate DNA right from your kitchen. So Ankita and Yogita, these are two very active students of our college, and they have been participating in a number of activities that go around in the college. And to apprise all of you, we have started a bridge program in our college where we have been doing experiments on because it's the COVID time and we don't have an access to laboratories. So we take up small experiments that can be easily done with the household material. You need not go out, and these have like we did the experiments on antibiotic sensitivity and resistance, isolation of DNA, isolation of human DNA for that matter. So from one of those sessions, we have Ankita and Yogita with us today, and they'll be guiding you how easy
it is to isolate DNA. So without wasting time, I invite Ankita and Yogita to take up this session, the charge of this session. Yes, Ankita.
Thank you, ma'am, for an amazing introduction and also for giving us this opportunity to be a part of this hands-on session on International Microorganism. We feel honored to be a part of this international event. I also thank each and every participant for being with us.
So without any further delay, let's begin with the session. I, Yogita Tomar, along with my partner Ankita Kapil, will be the speakers for today's hands-on do-it-yourself activity, which is about DNA isolation from plant cell sources, and that also at our homes using the things that are available at home.
We have students from different schools and colleges that have joined us today for performing this activity. So we hope that you will perform this activity along with us, and we also know that you are eager and enthusiastic to perform this experiment.
और इस एक्टिविटी के लिए जो भी मटेरियल्स रिक्वायर्ड है वो सबके घर में हर टाइम अवेलेबल होते हैं, इसलिए हमने ये एक्टिविटी चूज किया ताकि मैक्सिमम स्टूडेंट्स और जो भी प्रेजेंट है आज हमारे साथ पार्टिसिपेट कर सके और परफॉर्म कर सके.
So now we will begin with isolation of DNA. I am 100% sure that you all must be knowing about what is DNA and where is it located. If not, I will tell you in a very basic language. Every living organism in this world has DNA, चाहे वो plants हो, animals हो. अब ये क्वेश्चन अराइज होगा कि कहाँ पे DNA लोकेटेड है और DNA होता क्या है. तो हमारी बॉडी में मिलियंस एंड ट्रिलियंस ऑफ सेल्स होते हैं. उन सेल्स के अंदर होता है न्यूक्लियस. न्यूक्लियस के अंदर एक जेनेटिक मटेरियल प्रेजेंट होता है जो आपके पेरेंट्स से आप में, आपसे आपके ऑफस्प्रिंग्स में पास होगा. वो जेनेटिक मटेरियल होता है डीएनए, और डीएनए की फुल फॉर्म है डीऑक्सीराइबोन्यूक्लिक एसिड.
डीएनए में वो सारी इंफॉर्मेशन होती है जो आपको बनाती है, जैसे आपकी आईज का कलर, आपके हेयर्स का कलर, आपके हेयर स्ट्रेट होंगे, वेवी होंगे या कर्ली होंगे, स्किन कलर, सब कुछ ये आपके डीएनए में इंफॉर्मेशन होती है और यही आपके पेरेंट्स से आपको पास ऑन होती है. ऐसा नहीं कि सिर्फ इंसानों में या सिर्फ ह्यूमन बीइंग्स में ही होता है, इट्स प्रेजेंट इन एवरी लिविंग बीइंग इन द वर्ल्ड. सिमिलरली प्लांट सेल्स में भी डीएनए प्रेजेंट होता है और उसमें भी सारी इंफॉर्मेशन होती है कि प्लांट में कौन सा फ्रूट आएगा, कौन से कलर का फ्रूट आएगा, कौन सा फ्लावर आएगा, उस फ्लावर में कितनी लीव्स होंगी, कौन से कलर का होगा, वो प्लांट कितनी हाइट का होगा, एवरी इंफॉर्मेशन, सारी इंफॉर्मेशन होती है. और आज हम वही डीएनए अपने किचन में जो अवेलेबल प्लांट सेल सोर्स है उससे एक्सट्रैक्ट करने वाले हैं और बहुत ही सिंपल और इजी वे में. तो स्टार्ट करते हैं. अंकिता, रेडी?
यस योगिता, लेट्स स्टार्ट.
ओके, सो इस एक्सपेरिमेंट के लिए हमें जो भी मटेरियल्स चाहिए वो है. सो सबसे पहले वी नीड अ प्लांट सेल सोर्स. हम लोग बनाना यूज करेंगे, बट आप कुछ भी यूज कर सकते हो. लास्ट टाइम हमने टमेटो से एक्सट्रैक्ट किया था, आप कीवी से एक्सट्रैक्ट कर सकते हो, स्ट्रॉबेरी, पपाया, इवन अनियन से भी एक्सट्रैक्ट कर सकते हो. नेक्स्ट वी नीड अ टेबल सॉल्ट, जो घर में खाने वाला नमक होता है, दैट विल डू. नेक्स्ट वी नीड डिश वॉशिंग लिक्विड, जो घर में लिक्विड सोप होता है जिससे हम बर्तन वगैरह धोते हैं, दैट वी नीड टू यूज. एंड इफ यू हैव कलरलेस, अगर कलरलेस है तो वो बेस्ट है, बट अगर कलरलेस नहीं भी है तो कोई बात नहीं, सिंस हमारे पास भी येलो है हम ये यूज करेंगे. नेक्स्ट वी नीड मोर्टार और मिक्सर ग्राइंडर. यू कैन यूज ईदर. ओके, मिक्सर ग्राइंडर इज नथिंग, जो घर में मिक्सी यूज होती है जिसमें ग्राइंड करते हैं सामान, वो. नेक्स्ट वी नीड चिल्ड लिक्विड सैनिटाइजर. ऑल्सो मेक श्योर कि लिक्विड सैनिटाइजर हो, जेल बेस्ड नहीं. वी विल यूज लिक्विड एंड ठंडा होना चाहिए. सो वी हैड रेफ्रिजरेटेड दिस सैनिटाइजर ओवरनाइट. ओके. Next, we need clear small glasses, some spoons, 1 or 2 spoons, some bowls, and lukewarm water. Thoda garam pani. Okay, so we'll start now with the experiment. Okay, so सबसे पहले हमने जो भी plant cell source लिया है. Okay, so with the help of a kitchen knife or even with your hand, you have to make small pieces and put in the mortar. आप स्पून से भी और हाथ से भी छोटे छोटे पीस करके इसको मोर्टार में डालो और इसको अच्छे से हमें क्रश करना है. थोड़े से ही हो जाएगा. ओके, सो वी हैव टू क्रश इट नाउ. एंड आवर अल्टीमेट गोल, हमारा गोल है कि इसका अच्छे से पल्प बनाना है, इसमें बिल्कुल लंप्स नहीं रहने चाहिए, एक अच्छे सा पेस्ट, अच्छी कंसिस्टेंसी का पेस्ट बनाना है इसको, और अच्छे से इससे क्रश करते रहना है। और हम ऐसा क्यों कर रहे हैं, क्योंकि हमने अभी सुना कि डीएनए जो प्रेजेंट होता है वो सेल के अंदर होता है, तो डीएनए को बाहर निकालने के लिए हमें मैकेनिकल और फिजिकल थोड़ा सा डिस्ट्रप्शन करना पड़ेगा सेल का, ताकि सेल वॉल्स एंड मेम्ब्रेन्स ब्रेक हो, और ऐसा करने से 10% डिस्ट्रप्शन हो जाता है, बाकी 90% हम लोग आगे एक्सट्रैक्शन बफर बनाएंगे, उससे होगा। सो वी आर डन विद इट। सो इसको साइड में रखेंगे। नाउ वील मेक एक्सट्रैक्शन बफर। एक्सट्रैक्शन बफर को हम लाइसिस सोल्यूशन भी कहते हैं। लाइसिस मतलब तोड़ना, ठीक है, टू ब्रेक। तो हम ये एक्सट्रैक्शन बफर से जो भी मेम्ब्रेन, सेल वॉल्स, न्यूक्लियर मेम्ब्रेन्स है वो सब ब्रेक करेंगे। सबसे पहले हम इसके लिए लेंगे ल्यूकवार्म वाटर, गर्म पानी, अबाउट 20 ml, less than half of a glass, yeah. Then we'll add a half teaspoon of table salt, खाने वाला नमक जो घर में अवेलेबल होता है सबके। Yeah, to this we will add dishwashing liquid soap, about 2 tablespoons, 5 ml, थोड़ा सा, yes. And we'll give it a good mix. इसको अच्छे से मिक्स करना है। ओके, तो ये हमारा एक्सट्रैक्शन बफर रेडी है। अब हम इस एक्सट्रैक्शन बफर को जो हमारे मैश बनानास हैं उसमें ऐड कर देंगे। और हमें इसको बहुत अच्छे से मिक्स करना है, एंड बहुत स्लोली मिक्स करना है ताकि फोम ना फॉर्म हो। We have to make sure that there is no formation of foam. Okay? And the more we mix, जितना ज्यादा हम मिक्स करेंगे उतना ज्यादा डीएनए एक्सट्रैक्ट होगा, बिकॉज यहाँ पे हम जितना ज्यादा मिक्स करेंगे उतनी सेल मेम्ब्रेन्स एंड न्यूक्लियर मेम्ब्रेन ब्रेक होगी और डीएनए बाहर निकलेगा। Mix very well and also make sure that you are mixing it very gently. Yeah. So now I think we are done with the mixing. Yeah. So up now we'll filter the contents into a clear glass using a sieve. जो घर में आपके घर में छन्नी होती है जिससे हम चाय स्ट्रेन करते हैं वो यूज कर सकते हैं। Now जो अभी हमारे पास लिक्विड है, this has the DNA of the banana dissolved in it. इसमें हमारा डीएनए है जो हमने बनाना से एक्सट्रैक्ट किया था, वो डिसॉल्व है तो वो अभी नहीं दिख पाएगा। एंड इसके लिए हमें अबाउट 5-10 मिनट्स वेट करना है ताकि इसके जो कंटेंट्स है थोड़ा सेटल हो जाए, एंड उसके बाद हम नेक्स्ट स्टेप करेंगे। सो बाय द टाइम वी आर वेटिंग, आई विल टेल यू कि हमने एक्सट्रैक्शन बफर जो बनाया था उसमें हमने वो चीजें क्यों डाली थी, उनका क्या फंक्शन होता है, वो क्या चीजें कैसे ब्रेक करता है। ओके, सो फर्स्ट वी यूज्ड हॉट वाटर, गर्म पानी। व्हाई वी यूज्ड हॉट वाटर और कोई दूसरा मतलब नॉर्मल वाटर क्यों नहीं यूज किया? बिकॉज हाई टेम्परेचर हेल्प करता है प्रोटीन डीनेचरेशन में, प्रोटीन को डिस्ट्रॉय कर देता है, तो इसलिए हमने हाई टेम्परेचर वाटर यूज किया था। ज्यादा हाई नहीं, नॉर्मल ल्यूकवार्म वाटर विल वर्क। देन वी यूज टेबल सॉल्ट, घर में जो नमक होता है। नाउ टेबल सॉल्ट, दिस सॉल्ट इज नथिंग बट सोडियम आयंस। तो डीएनए में जो नेगेटिव चार्ज होता है, सोडियम आयंस उनको न्यूट्रलाइज कर देते हैं। एंड नेगेटिव चार्ज क्यों होता है? बिकॉज उसका जो शुगर फॉस्फेट बैकबोन होती है डीएनए की, उसकी वजह से नेगेटिव चार्ज होता है और वो न्यूट्रलाइज हो जाता है। ठीक, then we use dishwashing liquid soap. Now this is nothing but soap, detergent. Okay, so what happens, this helps to break down lipids and proteins in the cell and nuclear membrane. ये सेल एंड न्यूक्लियर मेम्ब्रेन्स को डिस्टर्ब कर देता है, डिस्ट्रॉय कर देता है। तो इसलिए वी एडेड ऑल ऑफ दीज थिंग्स इनटू द एक्सट्रैक्शन बफर, ताकि हमारे सेल की जो भी मेम्ब्रेन्स हैं वो सब ब्रेक हो जाए एंड द डीएनए कम्स आउट इन द सॉल्यूशन इन दिस लिक्विड। ओके, so I guess we're done with 3 minutes. We'll wait for another 2-3 minutes, and then we'll add chilled sanitizer into the glass. Now sanitizer we are using sanitizer because इसमें isopropyl alcohol होती है, about 90% isopropyl alcohol or even less, and it works. हाँ, तो क्या होता है, अभी लिक्विड में हमारा डीएनए डिसॉल्व्ड है, तो we need to precipitate it, जो होता है हमारा सैनिटाइजर डाल के, वो इकट्ठा हो जाता है, कोएगुलेट हो जाता है। Okay, so I guess we are done with 5 minutes, and now we can add the chilled sanitizer. और हमें इतना मेक श्योर करना है कि जो हम सैनिटाइजर ऐड करें उसके बहुत ग्लास की वॉल्स के सहारे हम ऐड करें along the edges of the glass so that we do not disturb the contents of the glass. Yes. So we have to now leave it for about 2-3 minutes for the precipitation to happen. And for the observation and whatever we now see, I'll hand over to Ankita.
As we are running short of time, so I will tell you the observations now. You will find a white layer of DNA. Why a layer? Because the DNA we took has many cells. Those cells must be having many nucleus. Those nucleus contains many DNA molecules. Those DNA molecules got entangled together to form this white layer. If you can see it by this video, or else I will now show you a presentation in that you can clearly see the layer. Is my screen visible?
Yes, Ankita.
So the first image you can see is the DNA we extracted using tomatoes. You can see this white layer. This is of the DNA we extracted using tomatoes. Another picture, that too of the DNA extracted using tomatoes. The third picture is the DNA that we extracted using bananas. In the fourth picture, you can see it very clearly that the DNA are coming out from the cells. We break down after mixing it well. Thank you.
That was lovely, Ankita and Yogita. Wonderful, wonderful.
And
I hope
बच्चों
को अच्छा लगा होगा DNA isolate कैसे करते हैं आपने सीख के आपको अच्छा ही लगा होगा
बच्चों
इसमें आपको जरूरत बहुत सारे इंग्रेडिएंट्स की नहीं है आपको सिर्फ नमक चाहिए घर के ग्लास चाहिए चम्मच चाहिए You can use a lot of ingredients other than they use banana. They have tried with tomatoes. You can go for kiwi. You can use strawberries. तो जो सीजनल फ्रूट अवेलेबल है यू कैन गो फॉर इट आपको रिजल्ट ऐसा ही मिलेगा थोड़ा सा वेरिएशन आएगा बट आपको डीएनए जरूर नजर आएगा so this the purpose of keeping this session was to just speak out loud that science does not stop at home आपको लेबोरेटरीज की जरूरत नहीं है आप घर पे भी कर सकते हैं घर के इंग्रेडिएंट्स के साथ और कोविड ने बहुत कुछ बुरा किया लेकिन सैनिटाइजर जरूर घर घर में पहुंचा दिया तो आप डीएनए जरूर आइसोलेट कर सकते हैं ना right so with that we'll be closing this session and now I would request Dr. Princey to please take over and we'll be starting with the quiz session now.
Thank you, Dr. Shelley and students. Well, very wonderfully demonstrated the DNA isolation. So now we come to the interesting session. Welcome to the quiz competition. That is the game of mind, intellect, interest, power, and joy. We know that you all are very excited and curious, but just hold on for some time. Let me just quickly tell you a few rules. So we have 2 separate quiz sessions for school and college students. Okay, each quiz has 15 questions and you have to attempt all the 15 questions and then submit it. So it's like a simple Google form. जैसे आप normally Google form fill करते हो, अभी पैंडेमिक की वजह से सबने अपने स्कूल्स में या कॉलेजेस में एग्जाम्स दिए होंगे यूजिंग गूगल फॉर्म सो इट्स लाइक दैट ओनली यू हैव टू एंटर योर डिटेल्स फर्स्ट योर नेम school name or college name depending upon whether you are a school student or college student and then you just have to choose the correct answers okay because it's a multiple choice question quiz so whoever scores best and submit at the earliest will be the winners so total 15 questions and 15 minutes only So without wasting much time, I'll just move to the quiz for school students first. So please pay attention. Only school students would be attempting this quiz. And I'm pasting the link for the quiz in the chat box. So hurry up. It's 12:40. You have time till 12:55. All the school students. So I repeat again, whoever submits first and scores the best would be awarded a prize. No, Anshu, undergraduate quiz, I'll be posting just 2 minutes. Let school students start, and then undergraduates are also going to have a quiz, but that's a different one. The level of questions is different for school students and college students. Just hold on for 10 minutes. We have quiz for college students as well. Ankit, the quiz for college students is for both undergraduate and postgraduate, so you can attempt that, not a problem. Not for teachers. Okay, so meanwhile, our college students are also very excited. I'll post the college quiz as well so that simultaneously all of you can attempt the quiz. Good luck. Hurry up, college students. We've got so many responses from school students, but no response from your end. Hurry up. Okay, I've sent the links again, but please be sure that you have time till 12:55 only. Any answers after 12:55 will not be evaluated.
So during this time, when our students are busy with the quiz, I take the privilege of talking to the teachers who have joined with us today. So, teachers, we as a group together, we have been organizing outreach programs with school students particularly, college students also. So we would like you all to please come forward and join hands with us. We'll be more than happy to help you out. In another 10 days or so, we are running an outreach program with the government senior secondary school at Shalimar Bagh, where we'll have students from 10th to 12th joining us for different science experiments. Now, because of this COVID time and because the school resources are not always enough for the experiments to be conducted. So we will try our best to give, though it will be online, but we'll try to give them a good experimental exposure. So if you all are interested, you can just drop us a mail and we'll be more than happy to associate with you. We are a big group and this is our motive. This is our initiation that we all have been working on. So, on behalf of all of us, I welcome you to join us. Professor Ruplal had an important assignment, so he had to leave the meeting. But he wanted us to convey this message to all our participants that we are open for all of you. For the poster presentations, we are still getting the entries, so probably we'll declare the results through mail.
Okay, last 5 minutes for you students. Hurry up.
हेलो हाँ कर दो मैं खुद ही सोच रही थी पता है मैं बोलना सोच रही थी फिर मुझे लगा क्विज का पोस्टर लगाया हुआ है तो वो कैसे लगा हाँ हाँ कर दो हाँ हाँ ठीक है हाँ, मैं बोलना शुरू कर देती हूँ आप ढूंढते रहो है ना मैं बोलना शुरू करती हूँ अच्छा ढूंढ लो हाँ अब हमें बिल गेट्स के लिए फिर से इनको कांटेक्ट करना है ना
Last 2 minutes, students. I'm very happy to share that some 55 students have attempted college quiz and
पुराना भी चल जाए तो भी चलेगा कुछ
only 25 from school.
हाँ
Hurry up, last 2 minutes. Nishant, quiz results will be announced at the end of this session and poster results, as Dr. Shelly mentioned, will be conveyed via mail because we are still analyzing.
शैली पे बोल पड़ी है और गौरी मैम भी बोलेंगी मेरे को शांति से कराने में अच्छा ठीक है हाँ इसके बाद हाँ सुकन्या मैम और चारू मैम मैं अभी बोल देती हूँ ना साथ साथ मैं बोल दूंगी आफ्टर द क्विज द पोस्टर विल बी डिस्प्लेड ऐसे बोल देती हूँ पोस्टर नहीं सर्कुलर ठीक है मैं बोल देती हूँ हाँ I'm back again. I need to make an important announcement to all of you. This coming November, from 15th to 18th, we are organizing an international conference on microbes and sustainable development. And there we have a special. We have more than 70 speakers from India and abroad who will be speaking on different aspects of microbiology. And apart from that, we also have a special session on science and society, which will be specifically focused on the students. So we'll have child-centric sessions under this theme. So we invite all of you for that conference to register. In a short while after this quiz, I would request Dr. Gauri Gadya to please present the poster of the conference so that everyone sees it. We can also share the link for registration and website in the chat box. It would be great if you all would join that.
Well, the time is over. So it's all done. Students, you should stop now because any entry after this will not be accepted. And I hope you all have enjoyed the quiz. The questions were quite easy. Some were tricky as well. So meanwhile, let's evaluate your answers. Moving forward, I request Dr. Gauri to share the brochure of the conference.
Once again, good afternoon to all. Now, here is the brochure and the circular for our upcoming conference in November. As you can see, it is in association with 7 Delhi University colleges and 2 of other universities, the Allahabad University and the Magad University. We have colleges associated: Acharya Narayan Dev College, Deen Dayal Upadhyay College, Gargi College, Kirodibal College, Metri College, Ramjas College, Sri Venkateshwara College, and also we have our partners, Fixgene Private Limited, Gurugram. I'll take you through the brochure now. So this is a 6th annual international e-conference of INSCAR Network for Indian Network for Soil Contamination Research, INSCAR. We would be having 70 speakers, as you have just known from Dr. Shelley. Now, of which about 30 are international speakers and 40 are our national speakers. We will have a range of speakers from various presidents of reputed international journals. And also we will be having speakers from various fields of microbiology, medical microbiology, clinical microbiology, environment microbiology, agriculture microbiology, and so on. So this would be a beautiful platform for those who want to become a microbiologist and get a glimpse of this microbial world. So we invite all of you to join this conference. There will be a special session on science and society, where we'll be talking of sustainable goals as discussed by Dr. Utkarsh Sood. And then we have beautiful workshops of scientific writing and communication, for computational biology, for metagenomic analysis, and a special workshop for college students, which will be training you in computational biology for the beginners. As Dr. Roshan said, bioinformatics is the science of today. When in COVID times, we can't do the practical work. So therefore, we present to you this small workshop in which you will get acquainted to the major techniques of computational biology. And from here on, you can take on to your master's and your PhD and other studies. Our last date of submission of abstracts would be sometime in October, but we are still open for registration. So please visit our website and you can contact us for further details. Thank you, Princey.
Thank you, Dr. Gauri. So I've actually pasted the link to the conference website in the chat box and also the registration link. So kindly visit and register for the conference. We are still open. So moving forward with our program, I would now request Dr. Kiran to kindly take over.
Thank you, Dr. Princey. Very good afternoon to one and all. As we have reached to the end of this event, firstly, I would like to invite Professor Sukanya Lal. She has joined Ramjas College as a lecturer and became professor recently. She has over 30 years of teaching experience. She has coordinated DBT Star College project in the Ramjas College for more than 7 years. Under DBT Star College, she has organized many hands-on training programs and skill development activities for students in collaboration with other departments and colleges of University of Delhi. Apart from this, she actively involved in organizing and coordinating outreach activities for promoting the science and education in rural areas at the graduations and school levels. She is also co-investigator in a multi-institutional research project on HCHY remediation. She has long list of publications in many reputed national and international journals. She is authored and co-authored chapters in books and also contributed e-chapters for undergraduate students. I request Professor Sukanya Lal to formally give conclude remarks of this event. Over to you, ma'am. Ma'am?
Yes. Thank you, Kiran, for beautiful remarks, and I must say expanded remarks for me and regarding my introduction. So just a minute, I will be starting it. So, good afternoon to all dignitaries and participants. I'm sure all of you excuse me. I am sure all of you are enlightened by the motivating and informative talks of eminent speakers who spared their valuable time in this program entitled Microsphere. I would rather say we are living in the megasphere where different types of microbes, plants, and animals are interacting in the surrounding environment, not only for their survival, but also for the survival of human race. Today's program was composed by Dr. Shelley, and she explained the theme Microsphere with high precision and enthusiasm. Principal Deen Dayal College gave an overview of the program and wholeheartedly supported this program, followed by a wonderful and heart-touching video of outreach programs organized by our team to inspire all the participants to give back to our society in one form or another. Professor Chandralekha Singh's motivated lecture to promote underprivileged students who either come from low socioeconomic background or are slow learners is highly admirable. She highlighted that it is our duty as teachers, parents, friends, and siblings to help those students to cope with their problems in a healthy way, and we should respect them. She further emphasized to promote gender sensitization and empower girls in the society to be successful human beings and to be successful in their professions. In addition, she also made us aware that we must let students know about our struggles while achieving our goals to make them capable to face their challenges in life successfully. It was indeed an inspiring talk. Professor Ruplal, on the other hand, has brought microbiology in the forefront and has also increased our responsibility to take up microbiology literacy mission on war footing. Because microbes have an important role in biodegradation, biostimulation, cleaning water bodies, and maintaining good health by maintaining good gut microbiome. This one-day conference is one such step in this direction. Importance of microbes was realized as early as the discovery of the microscope was very well documented and composed by Dr. Renu Solanki. Dr. Ashwin Kumar explained about the new upcoming field of using microbes in the construction material to reduce the cost of construction and to reduce carbon dioxide emissions during re-cement production processes. It's entirely a new concept to be materialized in the near future. Dr. Utkarsh Sood emphasized how microbes have benefited healthcare system by providing antibiotics, vaccines, etc., which are either the derivatives of the metabolic cycles of microbes or are the attenuated microbes. Antibiotics should not be used thoughtlessly to protect ourselves from multi-drug resistance. He also explained different parameters for sustainable development, which can be achieved only through microbial literacy. So I'm sure through today's international outreach program, you have witnessed wide-ranging benefits of microbes in our daily life. My colleague, Dr. Shelley, and all the team members have really put up the well-structured program in place in a well-coordinated way and in a very short time. I request them to continue this microbial literacy mission in near future to help mankind. I congratulate all of them. Thank you. Over to you, Dr. Kiran.
Thank you, Dr. Sukanya ma'am. And thank you for your encouraging words. Now I would like to invite Dr. Charu Dogra Rawat. She is an associate professor at Ramjas College, University of Delhi. She has more than 15 years of teaching experience. She is PhD in molecular microbiology from University of Delhi. She has postdoctoral research experience in muscle physiology at Jerry L. Pettis Memorial Veterans Hospital in California, USA. She received prestigious university faculty training fellowship to do MS in bioinformatics from University of Edinburgh, Scotland, United Kingdom. She has publication in internationally acclaimed journals and presented papers in national and international conferences. In 2019, she was selected as one of the young scientists to be part of 20-member delegations from India to visit universities in China to look for prospectors to develop collaboration in science and technology between the two countries. She has conducted many pedagogy workshops all over India and is master trainer at the Delhi Effective Education and Pedagogy Cluster under Delhi Research Implementations and Innovation set up by the Office of the Principal Scientific Advisor to Government of India. Apart from teaching undergraduate students, she has engaged in popularizations of microbiology, genomics, metagenomics, and microbiome in research areas. I request ma'am to acknowledge the participants and conclude the program. Over to you, Charu. Charu ma'am.
Thank you, Dr. Kiran, and a very, very good afternoon to all of you. Tell me and I forget. Teach me and I may remember. Involve me and I learn. This quote from Confucius philosopher highlights the essence of effective dissemination of knowledge, that is, emphasizing on inclusion of activity-based learning and principally apply what one has learned to solve the real-world problems. Microorganisms are already ubiquitously integrated. What is needed is to integrate microbial awareness, literacy, and utilization in our lives, be it in curriculum, teaching methodologies, lifestyle, or career. On International Microorganism Day 2021, MICROSPHERE, organized under the umbrella of INSCAR, has just tried to embark in that direction. Microorganisms are no longer remaining as a bioresource for lab research, but increasingly becoming more and more center of almost everything. Can we forget how a single virus has changed the face of the world? We somewhat witnessed this today, exploring the role of microorganisms as tiny contractors in lecture by Dr. Ashwini Kumar, or significant roles they play in attaining various sustainable development goals delivered by Dr. Utkarsh Sood. Microorganisms research has also opened up many avenues for setting up or working in a science-based companies, as emphatically outlined today by Dr. Amjad. Requirement is the identification of a role that can be of commercial importance. And then irrespective of social or economic background, the ideas can be incubated to eclose as a successful venture. Practical approach to learning exemplified by very well presented hands-on activity by two dear students must have ignited the spark to learn microbiology, as well as brought to the forefront how easily and with minimal, readily available things one can witness the fascinating aspects of science. Through session on careers in microbiology, Dr. Gauri and Dr. Roshan tried to remove the cobwebs, clear the blurry vision, and laid open various paths that can be tread. Quiz session, I am sure, would have tickled the brain cells with a dash of microbial knowledge and understanding. Through a plethora of in-house activities and outreach programs, we, under the able guidance of exemplary visionary Dr. Roop Lal, aim to make visible the invisible by tweaking the teaching learning process of microbiology. I hope you all had an enriching experience. Thanks and my best wishes.
Thank you, ma'am. I now would like to invite Dr. Jasbinder Kaur. She is an assistant professor at Gandhi College, University of Delhi. I request Dr. Jasbinder Kaur to give vote of thanks.
Thank you, Dr. Kiran. Good afternoon, everyone. As aptly put by Ed Yong, the famous writer of I Contain Multitudes, I was struck by how different everything seemed with microbes in mind. With that quote in mind, I would happily take this opportunity to announce the quiz winners now. So for the college students, it's a tie between all the four students. Miss Harshita Raj, Kirorimal College. In no particular order, as all are tie and all have got full marks. Congratulations. Mr. Harsh Garg, again, Firodimal College. Anisha Raj from Miranda House. Ishita Devna from Gandhi College. My colleague, Dr. Princey, would be typing in these names in the chat box for all to see. Now coming on to school winners, again in no particular order, all have got a tie. Sakshi Verma, Army School, Jammu. Janvi Gupta, Maxford School, Rohini. Natasha, KR Mangalam World School, South City. Bhavya Gopakumar, Mary Nilliam Senior Secondary School. So congratulations everyone once again, and you will be contacted later to share bank details to transfer the prize amount. And for poster winners, you will have to wait as results are still being analyzed, and results would be mailed to the winners in next 2 days. With that, I take this opportunity to end this program with a vote of thanks. We are all overwhelmed with gratitude to each one of you who chose to be with us for this celebration of the tiny life around us. Words of gratitude won't suffice for our eminent speakers who took out their precious time and presented their talks. We pray that you all keep well and keep shining your knowledge onto the world. We are immensely grateful to all the esteemed principals and directors of organizing and associated institutes for their steadfast support. We are equally grateful to the entire organizing committee members for being part of this event, the technical support team, and last but not the least, to all the lovely participants without whom this event would not have been a success. This event was a minuscule attempt to actively facilitate and foster the kind of transformation that leads us all to new doors. I pray to Almighty that we all keep well and carry forward at least some of what we have learned in this event. Thank you and have a nice day. Over to you, Dr. Shelly, for a picture maybe. Thank you.
Thank you, everyone. Thank you. So, can we all say a goodbye to all our dear participants together? Students, it would be great if you could switch on your videos and we can just have a click.
No.
No? Yeah. Just give me a moment and just clicking. Yeah. Thank you so much, everyone. Thank you for joining us today and making this event successful. Thank you to the Microbes also who gave us this opportunity to celebrate. Thank you.
Thank you,
Thanks to
Shelly.
the organizing team. Thank you, Dr. Shelly.
Thank you,
It was such a great
Thank you,
program.
Shelly.
Wonderful.
बहुत ही बढ़िया प्रोग्राम हुआ, बड़ा टाइम से हुआ.
Thank you, everyone.
And so much of labor and so much of lectures, everything went so well.
And it went very coordinated. That's
Yes.
to be appreciated. Without a single glitch.
It's all because of you. Thank you so much.